Great. All right, great. Well, thanks everyone. We had some great talks in the last session. We're going to move on to our late morning session on status and trends in nutrient studies. And Janis Cooke is going to kick us off with our opening remarks, and she's also going to be the moderator for this session.
All right. So thank you everyone in the room and online for coming back. I'm Janis Cooke with the Central Valley Water Board, and I'm also a Delta RMP nutrients TAC measure member. So I'm a fan of the public radio program. Wait, don't tell me. And the host Peter Sagal has this line of, we have a great show for you today. And so that's what I feel like about this next session. For the next couple of hours, we're going to delve deeply into status and trends in our nutrients studies. This is work that has been done partly supported by the Delta RMP and also done alongside the Delta RMP. So thank you for all those collaborations. We're going to be talking about, hearing about nitrogen and phosphorus forms, cycling, and also compartments of the ecosystem influenced by those nutrients. So specifically, phytoplankton species and abundance, and also harmful cyanobacteria blooms. This is work that directly fills gaps in the Delta Nutrient Research Plan, and it also sets us up for what are some of the next questions we have for the future. So thank you to Dylan and Lisa and Tamara and Keith and Ellen for your presentations. And first up, we have Dylan Stern from a project manager from the Delta Stewardship Council and the Delta Science Program. So
thank you, Janis. An obstacle course.
Yeah.
I like your style, Tim. I'm going to copy it.
Works for me.
All right. Wonderful. Thank you so much for having me today. It's really an honor to be here. I feel like a lot of you are close friends and family at this point. My name is Dylan Stern. I'm a program manager for science funding at the Delta Science Program. And today I'm really going to be giving a Delta Science Program perspective. So, it doesn't cover the whole universe on progress that's been made to address nutrient data gaps and that specifically have been identified in the Delta Nutrient Research Plan. And also the Science Action Agenda, which is the Delta Science Program's sort of guiding document for all of our funding decisions. Next slide, please. So what's the deal with nutrients in the Delta? I mean, this has already been sort of covered, but it's a weird dynamic that keeps me interested in coming back for more. Nutrients are not a limiting factor for primary productivity in the Delta. We have plenty of nutrients, maybe not anymore. The mostly we have anthropogenic sources, wastewater treatment plants and agriculture. And yet we do not have the typical symptoms of eutrophication in the Delta. And we have generally low levels of phytoplankton. One thing I've learned is that nutrients are a critical piece of the puzzle, but they are not responsible for the full puzzle, nor are they the only variable that matters in the food web. So we must consider abiotic factors such as temperature, salinity, and light, and consider the different forms, sources, and complex transformations that occur. And also, what is the optimal amount of water times the concentration of nutrients to get the nutrient load? And that's critical information for sort of determining the appropriate regulatory limit if we need one at all. And so hence the foundational Delta Nutrient Research Plan question, is there a water quality problem and are nutrients contributing to the problem? And sort of, I'm going to talk about the progress that's been made toward answering those questions, because I'm not convinced that we do know that the nutrients are a problem and that they are contributing to the problem. So I think it's a good question for us to keep talking about. And the reason that we care obviously are the effects that are linked to nutrients, harmful algal blooms and their toxins, non-native invasive aquatic macrophytes, low dissolved oxygen, and low phytoplankton. Next slide, please. So just a slide on what I'm going to talk about today, a little brief history. I want to mention the Delta nutrient stag and the set of studies that we call Operation Baseline, sort of funded by the Delta Science Program. There's a phase 1 and a phase 2. And then we've also funded some competitive nutrient related work that I'd like to talk about and sort of link it all to the science action agenda and a progress summary that was created in support of the next iteration of the science action agenda. And then sort of talk about future work and plug the HABs workshop that's coming in. So next please. So a lot of this was covered already, I guess. But I think to start us off, back in 2010, before it was even really in the system, the Regional Sand Wastewater Treatment Plant was issued a new permit in 2010 that included ammonia and nitrates due to emerging science about the effects of excess ammonia on phytoplankton, the base of the food web. Jumping ahead to 2013, the Delta Stewardship Council adopted the Delta Plan, management and regulatory plan for achieving co-equal goals of improving water supply reliability and the Delta ecosystem. And that Delta Plan has a recommendation for the Central Valley Water Board to develop a study plan for nutrients in the Delta. And the regional board accepted that recommendation and has obviously completed and gone through with that as well. So then the strategic plan was created, which includes direction to develop and implement a nutrient study plan for the Delta. And then regional board staff and the Delta Nutrient Stakeholder and Technical Advisory Group, Delta STAG, Delta Nutrient STAG. Has it been renamed? No, okay, Delta Nutrient STAG. Completed a series of white papers, convened a whole series of meetings with subgroups, compiled research recommendations from white papers, and then developed a really robust process to prioritize the recommendations. And then in between 2015 to present, the Delta Science Program has funded a number of studies that are related and sort of guided by the nutrient plan, nutrient research plan, and the science action agenda. And I will be going into more detail on that. But then also noting that the regional sand wastewater treatment plant upgrade is complete at this point for the nutrient portion. Next slide, please. So I've had the honor and pleasure of attending quite a few of the Delta Nutrient Stag meetings, and I've observed the process. It's been led heroically by the Central Valley Regional Water Quality Control Board. Janis and Christy really did an amazing job guiding the process. And I feel personally that it's been really successful, and the group has really accomplished a lot. And so Tom already covered the specific papers that were created, but they were really foundational, and everyone should read them and get a copy of them. Okay, next slide, please. I just want to mention that this has been an extremely collaborative process, and my template only had room for 10 logos, but there are more people involved than this. But I just wanted to give a shout out to some of the funders, state water contractors, regional SAN, Delta RMP. I should have had Delta RMP on there. Can't believe it. Sorry about that.
Yeah.
And then a ton of researchers that we've worked with, including USGS, Cal Maritime Academy, SFEI Aquatic Science Center, Virginia Institute of Marine Science, and DWR and UC Davis. So yay, thank you all for for your work on this. Next slide, please. So in 2016, the Delta Science Program was really interested in pursuing answers to a lot of these nutrient questions and wanted to jump right into the science and fill all the information gaps and measure everything. But resources are always a limitation, and the data and information gaps are really vast. And so first, next slide, please. First off, we wanted to evaluate potential hypotheses related to the wastewater treatment plant upgrade and the massive reduction in nutrient loads that were expected from that, and sort of then figure out what studies needed to be done to sort of answer those hypotheses, research plans and data gaps that could be filled for the purpose of studying the upgrade, but also with broader implications for the entire system. So what we're really talking about here is the Delta Nutrient Research Plan question of what are the gaps in our understanding of the problem, including status and trends. Because we have the Nutrient Research Plan, and it's like really large and it's great. We needed to get more into specifics. And so this really great effort led by Dave Sen, Tamara, a ton of other people, we wrote this conceptual framework that looked at different tiers of responses and what potential scenarios could occur for different endpoints, phytoplankton, even up to fish. And so the Delta Science Program hosted a workshop in 2018. I think some of you were actually there. This is it right here. And so we brought together scientists and managers to present the draft conceptual framework. And we asked participants to weigh in on how feasible, where'd it go? How to best link ongoing and future research with management needs. So the targeted discussions really talked about the importance, the relevance, and the feasibility of future research related to these topics. And everyone at the workshop really agreed that maximizing coordination and analysis across various entities and approaches, including monitoring, special studies, modeling, was paramount and extremely important. Next slide, please. Coming out of that workshop, this graphic has the gory details of data and knowledge gaps, their relative priority for evaluating responses to the nutrient reduction expected after Regional Sands Eco Water Project was completed, which was expected to significantly reduce inorganic nitrogen and has. And I'm going to come back to this later, but it's a lot, as you can see, a lot. And I could get into the weeds, but then I wouldn't have time to talk about anything else. So let's go to the next slide, please. So the studies that we pursued, that the Delta Science Program pursued in 2017, were exciting and experimental, and sought to develop new methods that could eventually fill Delta Nutrient Research Plan data gaps, like high-frequency fluoroprobes to measure phytoplankton taxa. And the second picture is the benthic flux chamber, also known as the sediment Roomba, which was able to measure, it was actually able to measure the nutrient sediment flux, and it's just mind-blowing, and I love it. And had some really interesting results that I'm not going to talk about. And so the Delta RMP, the Delta Science Program, state water contractors, has already been mentioned, but have supported these high-frequency boat-based measurements of nutrients, water quality, phytoplankton, and now cyanotoxin data, essentially taking a snapshot of nutrient concentrations and other water quality parameters at the delta scale, which was so necessary as part of our status and trends effort. And so another ultimate goal of that data collection is to get information that can be usable for these process-based modeling, where we can actually get at a lot of our management questions. And this modeling work, which you will hear about later, was collaboratively funded across many different folks. And in 2017, the science program contributed toward the SFEI project to develop the biogeochemical model that's linked to the hydrodynamic model. And so I don't need to talk too much more about that, but a lot of amazing progress has been made in that arena as well. Next slide, please. And then I have a bunch of slides about Operation Baseline. Our executive officer, Jessica Pearson, termed this phrase Operation Baseline when she heard about all the studies that we were planning, because we were trying to establish a baseline before the major management change occurred in the system. And she thought, and it was very catchy, so we went with it, and it has stuck. And so, yeah, so several of the studies I'm going to talk about briefly. We funded a study collecting high frequency measurements, as I already described, and this is sort of looking at like what are the status and trends. What are the most important processes that transform nutrients in the delta, and what are the rates at which these occur? And so these high frequency measurements are very helpful in trying to answer those questions that are identified in the Delta Nutrient Research Plan. Next slide, please. Another set of pilot studies ambitiously examined nutrient transformation and the ecological responses. So talking about phytoplankton, zooplankton, aquatic vegetation, within and across several wetland in habitat in the Delta and Sassoon area, and additional support was provided by state water contractors. And so this was a coordinated sampling effort across water quality, phytoplankton, and zooplankton, and it was very successful. And it was sort of a demonstration that this type of sampling can actually occur. And related to the nutrient research plan, looking at what are the spatial temporal trends in nutrient-related effects in the delta, especially phytoplankton production, and then looking at how do nutrient concentrations, loads, and cycling affect the growth of aquatic macrophytes, because aquatic macrophytes just haven't gotten a lot of love. Next slide, please. And then in 2019, the Delta Science program invested in phase 2, which was meant to really implement a lot of the new methods that were just developed, and take it a step further and collect more data. The upgrade had not happened yet, and there was still more data to collect. Next slide, please. So this set included a lot more both-based measurements with the USGS team. We also funded a synthesis effort that fed into a modeling effort. This time, working with DWR and VINS, Virginia Institute for Marine Sciences, and looking at a modeling effort with the schism model that focuses on the Cash-Loo complex and has an aquatic vegetation component to really look at patterns of productivity in the delta along the zone of influence of the wastewater treatment plant. And so that, and then the third project there was actually getting everyone on the same boat to, not literally on the same boat, to measure picophytoplankton, the tiniest of tinies. That was a big data gap. We don't know how, not usually collected, we don't know much about them. So we, that was a big multi-agency effort. Thank you, Ted. And so, now we're, leveraging existing monitoring programs and adding this extra analysis to get a more holistic look at the phytoplankton picture. Okay, next slide, please. And then we held a competitive proposal solicitation in 2018 in combination with CDFW's Prop 1 program. And related to nutrients, the Delta Science program awarded this project called Assessing Sediment Nutrient Storage and Release in the Delta. And so this was a USGS and UC Davis project. And it was really all about nutrients in the sediment and the microbial community that's associated with it. And then the effects on harmful algal blooms, aquatic vegetation, and drinking water quality. And so the study investigated the amounts and types and dynamics of nutrients in the delta sediments. And so there were cores taken, and the microbial community was examined, as well as nutrients. So the results are really meant to determine how the reduction in nutrient inputs will affect the sediment nutrients and microbial communities. And so this is a map of different microbial groups, actual data from that project. I wish I had time to go into it. And also the hope is that this data can be included into modeling efforts as well as sort of a missing piece of the puzzle, you know, the interaction between sediment, microbes, and the water column. Next slide, please. And more recently, in 2020/2021, USGS was, and Tamara and Keith are on this project. It's called Harmful Algal Blooms and Cyanotoxins in the Delta, Occurrence, Distribution, Trends, and Environmental Drivers. So noting this shift toward harmful algal blooms, which has just like emerged as a major problem. And so there's this recognition that we don't have enough information spatially about cyano-HABs. So to address the data gap, the study has added cyanotoxin sampling to water quality monitoring stations and on the boat-based measurements, and adding more sensors that continuously measure the abundance of 4 different phytoplankton groups that I mentioned earlier, the fluoroprobe, to identify eventually temporal patterns and key environmental drivers of cyano-HABs and the cyanotoxins. And so these are linked to these questions from the Delta Nutrient Research Plan, you know, the spatial temporal trends related to cyanobacteria blooms, toxins. It just really hits the mark. And then the relative importance of nutrients versus other factors in promoting cyanobacteria dominance and or cyanotoxin production in the Delta. Okay, next slide, please. So back to this lovely figure with the data gaps. I just highlighted in blue, sort of at a high level, what the studies that the Delta Science Program has funded in coordination with many other funding entities, but some of the progress that has been made sort of hits a lot of these data gaps. And so, you know, I think we still have data gaps with aquatic vegetation, the phytoplankton primary productivity rates, and then, you know, microbes don't really get enough love either. So, you know, there are still data gaps, and these projects don't 100% cover these data gaps, just to be clear, but they do make a lot of significant progress and or establish tools that can help us continue to collect this data. Next slide, please. I mentioned the science action agenda. The studies that we funded were based on the very first science action agenda, which covered 2017 to 2021, and specifically this one science action to implement studies to better understand the ecosystem response before, during, and after major changes in the amount and type of effluent from large point sources in the Delta, including wastewater treatment facilities. And so that's really the nexus to the science action agenda for this work here. And what they found was significant progress, 19 different science actions. And this was a collaborative process. We published this online. Thank you. And asked everyone for their feedback. You know, do you agree? Has there been significant progress? Did we miss any projects? And some of the feedback was up here, but you know, it's really important to measure the ecosystem response. And we need to examine the stressors on benthic and detrital food webs, develop response curves, and then a lot of barriers were mentioned about funding and coordinated monitoring. And then the data need identified here was continuous data for inorganic nitrogen, which just like aligns with everything that we've been talking about. And next slide, please. Can you go back one?
Sure.
Okay, next. So now definitely talking about the future here. Next slide, please. We have a new science action agenda, and I think a lot of you were involved in the very highly collaborative process. And the science action agenda is really meant to let us as a science community tackle the complex issues in the Delta that span many different topic areas in a rapidly changing system with limited resources, right? And so thinking about what are the highest priority things across the whole Delta system. And so it prioritizes and aligns science actions to inform management. It's collaboratively developed and identifies major gaps in knowledge, not unlike the process that we went through with the conceptual framework and the Delta Nutrient Research Plan. But it's a really important to develop those very specific topic-specific plans as well. Next slide, please. So yeah, a lot of text on the slide, but I just wanted to highlight the science actions and management needs and management questions that were developed as part of this new science action agenda process, and sort of highlight the most important ones. So science action 5C is determine how environmental drivers, including nutrients, temperature, water residence time, interact to cause harmful algal blooms in the Delta. Identify impacts on human and ecosystem health and well-being, and test possible mitigation strategies. And so the environmental drivers, the human and ecosystem health effects and well-being impacts are in dire need of investigation. And effective management strategies of HABs and their associated toxins is an area in need of science support as well. And I don't need to read the rest of these. Check out the link to the science action agenda if you want to look at the rest of those. So sort of looking at a high level on the Delta Nutrient Research Plan, gaps and information needs, the questions, and this is my opinion, okay? I feel like there has been significant progress in the status and trends. Is there a water quality problem? But I don't think we have an answer yet. I don't think we know if it is a problem. I think in some cases it is, and it definitely is a contributing factor to harmful algal bloom development, severity, and all of that. And not that it's not important to pursue, but anyway, I just think we have a lot more to look at. And then are nutrients contributing to the problem? Also, some progress here. Existing needs are really focusing on aquatic macrophytes and the nutrient transformation rates. And can nutrient management help address or ameliorate the problem? Again, some progress here. And I just want to stress the need to continue to prioritize the spatial temporal trend data collection for a holistic look at nutrients. And then, there's been a lot of progress made in the data collection and setting up modeling efforts with the hope that that will allow us to look at the management implications. What can we do about it? So I feel like there's a lot less progress in there, but we're very close to that point of being able to tackle those questions with all of the work that's been funded by everyone I mentioned.
Close.
Yes, very close.
All right.
Okay, next slide. More synthesis, more data. HABs workshop. Next slide, please. I think that everyone here realizes that monitoring harmful algal blooms is critical to protect water quality and public health, but there's no comprehensive, program or strategy or funding. So the Delta Science Program is hosting this 2-day hybrid workshop focusing on where HABs monitoring and data collection can be optimized to create a strategy for the Delta. And I think Central Valley Board has been working closely with the Delta Science Program on this, and I encourage you all to attend. It's either in person or via Zoom. And that is it for me. Thank you so much.
I feel like we need like your third to last slide with the Delta RMP and the Delta sites program. We need it over there on the bullet,
Yeah.
projected over on the bulletin board. So,
We can make that happen.
all right. So, we're at time for Lisa's talk, so I think we'll hold questions
Okay.
for the next, but thank you for giving us up in this session. All right, next up, we have Lisa Thompson, senior scientist with Regional SAN, talking about some of the really intriguing work that she and Tim have been doing in the past 3-4 years. Lisa. What do you know?
I'm just gonna keep saying next.
Okay,
It'll either be click or a slide.
okay,
I'm just gonna say next.
sounds good.
Alrighty. Cool. And is that gonna turn and look at me?
She's working on it.
Okay, so I'm just gonna start that. We'll see where we go. Okay, so good morning, audience in the room. Good morning, everybody. Hope all the cameras coming over. It's very scary. Hi, everyone in TV land.
It's like, yeah, opposite of what.
So I'm going to be talking to you mainly about one big study that was funded by the Delta Regional Monitoring Program and also a number of other groups and a big bunch of collaborators. It's definitely not the Tim and Lisa show. But I am going to refer to some earlier work as well that I personally was not involved in, but really led to this work, just to kind of put it in perspective and show you how one thing does kind of lead to another, and we are making some progress. Next. So first off, the Sacramento River Nutrient Change Study, you can see by the collaborators here, we had a large group of collaborators. That was necessary because we tried to look at so many different pieces of the ecosystem. We needed the expertise. Lots of logos up there, but I won't dwell on that. Next. So I just wanted to step back though and give you that perspective on why we did what we did here. So this graph here is from a study that Tamara Kraus was the lead on. It was funded by the Interagency Ecological Program, huge number of collaborators, a lot of people in the room here were involved. And what they were looking at was what would happen if the wastewater treatment plant, Sacramento Regional Wastewater Treatment Plant, went into what we call diversion and didn't put wastewater in the river. And they were able to do that for about 30 hours. And they looked at what happened in the river. They had boats floating down the river with a parcel of water that had the nutrients and another parcel of water that didn't have the nutrients. So back and forth between these parcels. And so the graph is showing you on the y-axis the chlorophyll. So how much algal biomass do you have? The x-axis is the distance upstream and downstream of the treatment plant. So there's a black vertical line here. That's where the treatment plant is discharging. And the take home, if you look at all these dots, is they're all falling on the same decreasing curve. The dots that are shaded are when there was wastewater in the river. The dots that are open is when there was not wastewater in the river. They did this 2 different times. So they did it in the spring and the fall. Take home is the chlorophyll just went down either way. So whether or not you had ammonia in the river from the treatment plant, the chlorophyll was going down. Now, the last little bit is kind of speculation. We're still figuring this out, but some reasons that have been suggested for why the chlorophyll are so unhappy out there is the river is narrow, it's channelized, it's deep, it's supposed to scour so that you have a nice channel for ships to come up. Deep and narrow and dark is not necessarily a happy place for phytoplankton. And phytoplankton experts have looked at the phytoplankton in the samples and went, good grief, they're kind of exploding and dying and they're really depressed. What's going on out there? But from what we can tell, it's not the ammonia killing them because they're looking depressed even without the ammonia. So one of the things that came out of this is we thought, well, still don't really know if the nutrients would make a difference if you had conditions where the phytoplankton weren't so unhappy. And there is, in fact, a phytoplankton happiness monitor, but I'm not going to talk about that today, but you can test that. Talk to Minnieberg or some of the other phytoplankton experts. So what we started looking at was where else in the Delta could we go that wasn't so deep, wasn't so dark, wasn't so fast flowing, where the phytoplankton might have a chance would grow better. And then we would look at what happens with and without nutrients. Next. So the objective of this particular study is multifold, but the main thing is looking at as much of the food web as we could without going all the way up to fish. What's going to happen when you take the nutrients from the treatment plant out of the system? And we were doing this study prior to the biological nutrient removal upgrade. So at this time, it was ammonia that was going into the river, predominantly from the treatment plant. We were able to do a 40-hour wastewater diversion thanks to some of the other work that was going on in the plants, a little bit longer time than was done in that previous experiment. And we decided to go over to the East Delta, where the channels are shallower and the flows are slower. So we'd hope that there would be better conditions for phytoplankton. And then we would try to measure as much as we could and see if things made a difference. Next. So here's our study area. And up at the top here, I'm going to point for the people in the room. So we've got the treatment plant up here near Freeport, the Sacramento River coming down. And then in blue here, the Sacramento River keeps going over towards Isleton and Rio Vista. So that's the area that was studied in the previous study. But down at the bottom, you can see these 3 purple lines. Those are the side channels that we decided to study for this project that are shallower and slower flow. So there's Janis Slough, North Fork McCollum River, South Fork McCollum River. All the green dots are places that we sampled, and I'm mostly going to be focusing on the sampling sites that are actually down in those 3 side channels today. The purple lines are where the USGS high resolution mapping boat was out sampling. And they did that each of the days during the study. So we have a big picture view of what was happening, and then we have grab samples. So we've got more specific data. Next. So here's our conceptual model looking at the system. It's the same map, but now we flipped it on an angle. You've got the Sacramento River up at the top and the back. The city of Sacramento and the treatment plant are up there at the top. The 3 side channels are kind of blown up at the front, left to right, and then there's 3 side sloughs that come into the South Fork McCollum River, and that's a whole other neat story. We won't have time to go into that today, but might be worthwhile for future work. Next. So up at the top there in this current loading condition, that's current as of 2019, we have predominantly ammonia coming into the river from the treatment plant, a little bit of nitrate, and then there is nitrate coming from upstream from other sources, urban runoff, agriculture. I'm not going to pick on people, but there's other sources out there. Next. And so what you'd expect to happen when you get down to these channels is by the time the water gets down there, that the ammonia would have partially nitrified. And so you're starting to see greater concentrations of nitrate, but you're also seeing some ammonia. Next. Okay, so then what do we predict is going to happen if you take the treatment plant and kind of take it out of the picture? Next. So up top, now we've got our treatment plant sitting there, but no nutrients coming in the river. Thanks. And what we'd expect to see downstream is some nitrate, no more ammonia, because it's really only coming from the treatment plant. And what you're really seeing there is background ammonia from the rest of the system. Next. And the flows are slower, so the little clocks tell you the time. So if the Sacramento River is 2 o'clock, we're seeing 4 o'clock to 8 o'clock in these side sloughs in terms of how long it takes the water to move through. And also because they're shallower, we're expecting greater light penetration. This is not really penetration, but we're just expecting the light can get to a greater proportion of the water column because the water column is shallower. Next. So we'd expect if the nutrients are what's really making the difference, and we put the phytoplankton in an area where there's enough light they could be growing with nutrients, we take the nutrients away, we expect to see fewer phytoplankton. That's kind of the Mickey Mouse version, and I can tell you now, it turned out to be more complicated than that. But anyway, that's what we predicted. Okay, next. So because this is only a 48-hour experiment, we're expecting in the case of zooplankton, they can't produce and they're not going to die off that fast. So we're not expecting them to change, but we might see changes in growth. And Wim Kimmer and his folks at San Francisco State University were on board to look for that. Next. And we're going to look at clams and how much grazing they might be able to do. We weren't expecting clams to change abundance over the time frame of the experiment, but Tim Musson and the clam people and many, many interns counting clams were able to get us clam abundance data and grazing rates. Next. All right, so another way to look at this is to look at food web diagrams, which give you a little better idea of abundances. On the left side is the Sacramento River system, and on the right side is what we think we'd see in the side channels. And then the far left is current wastewater loading, and then I guess it would be the second panel for the people at home, no wastewater loading, and then under the side channels header, you've got current and no wastewater loading again. So on the left side in the Sacramento River, because we think it's too dark for phytoplankton to do much of anything, click. It's there. Oh, no, we'll go back. I didn't, the clicks are later. Okay, let's go back one. Okay, anyway, you expect that even if you take out the ammonia, you're not going to see any change in phytoplankton because they weren't doing anything with the nutrients anyway. But over in the side channels, you might expect to see more phytoplankton in the first place because they've been able to use the nutrients. If you take the ammonia away, you might expect to see less phytoplankton, but you don't expect to see a change in zooplankton and clams over a 48-hour period. So we're really going to be focusing on whether or not we see a phytoplankton response, but we will look at the other parts of the food web just to kind of get a handle on what they're doing. Thanks. So off we go with the experiment. And so the neat thing was at this time of year, the treatment plant nutrient loading at that time was making up about half the loading in the river. So by turning the treatment plant essentially off, we got rid of about half the nutrients. So the blue part of this bar on the left is the ammonia from the treatment plant. There's a really thin line that represents the nitrate that the plant used to discharge. The purple is the upstream dissolved inorganic nitrogen, and the orange is the upstream dissolved organic nitrogen. So with the treatment plant going into diversion, what we had was just the upstream nutrients left in the system. So pretty substantial reduction in nutrients, but still some nutrients in the system. Okay, so very quick slide about the methods, just to kind of give you an overview of what we did, but we had flow modeling, so we had a sense of what tides and inflow river inputs would be doing to the system. We want to know where the water is coming from, because we do have McCullamy River water as well as Sacramento River water. The USGS boat doing the high resolution mapping, water quality sampling and lab analysis by the Regional Sand Environmental Lab, plankton enumerations by BSA Environmental in Ohio, carbon uptake coordinated by Minate Berg at that time at Applied Marine Sciences and now with the Geological Science Associates. Zooplankton growth by San Francisco State and Wim Kimmerer, and then clam collection analysis by Tim. And so up top is the USGS Landsteiner boat. That's the boat that did the high resolution mapping. The other three boats are put together from Regional Sand, the USGS, and San Francisco State. And each of these boats had a channel that it focused on. And the neat thing, though, is that the people on the boats are from all the different organizations. So on each boat, we had this phytoplankton specialist, a zooplankton specialist, a water quality specialist. So the teams were mixed up from the different organizations, but focused on a different channel. Next. Okay, so now getting into the results, and this first one is for ammonium. I'm going to dwell on this one for a little bit because it helps you see what happened with the experiment. It's a really nice overview. So on the far left is the first day of the experiment. At about midnight, 1 a.m. on September 10th, the treatment plant operators stopped the effluent valves and nothing was heading out to the river. And so by the time we were out sampling, there was already some wastewater, free water moving down the river. So that's the blue line. Down in our main study area, you can see the colors are still warm, kind of oranges and yellows. So down in that area, we still had the wastewater from the treatment plant. We're just kind of rolling into our experiment here. On the second day, the mapping crew is focusing just on the downstream area, but you can see that it's all mostly turned blue. So now that wastewater, free water has moved down into those side channels, and we're able to start looking at what's happening without the wastewater. On the third day of the experiment, we're still in our main study area, showing these cool colors. So the nutrients from the treatment plant are pretty much all flushed out of that system. But you can see that we're starting to see this orange line starting from the area of the treatment plant and moving down. So because it was only a 48-hour wastewater hold, about midnight 1 a.m. on September 12th, the treatment plant operators opened up the effluent valves and the effluent is starting to go into the river again. So you're starting to see ammonia coming down. And we just finished up our sampling before any of the wastewater started making it down to the system. So it worked out. The timing worked out astonishingly well, considering the scenario. But the neat thing here is you can see that there is a pretty dramatic change in what's happening with the ammonium. So that first step of did we actually create the experiment we hoped to create? Looks like we did. Yes. Right. So now we're looking at nitrate, just to look at another form of nitrogen and knowing that there's background nitrate in the system as opposed to very much background ammonium. So we still had a really nice pattern. So you can see warmer colors down here on this first day where you've got various sources of nitrate there. And on the second day, when you've removed any possible nitrate from the treatment plant, it's getting cooler, and then even more so. You'll notice that in the South Fork McCullamy, the colors are a little bit warmer. The flows there are slower. There's a lot of mixing with these side sloughs that are kind of shown in gray off to the right. And that makes the South Fork McCullamy really interesting for future study, but not necessarily the most perfect place for this particular study, but it still worked. Okay, next. All right, so now we're starting to look at responses, and this is chlorophyll pigment. And the take home here is that really was not much change across the 3 days. You can see more activity in the South Fork McCullamy, a little bit more warm colors happening there. It's shallower. It's got these side sloughs, probably pulsing in and out. And actually, it was interesting. I think on this one of the slides Tim showed, it was catching my attention when he showed the slide. I know there's a million, you won't remember them, but it was a heat map slide of how old the water was. And these side sloughs were really red. The water sits in there a long time. So if phytoplankton can grow, they'll grow. They might even use up the nutrients there, but it's some of that may be pulsing in and out of the South Fork. Anyway, at least, we see here with chlorophyll, not much of a response, which wasn't really what we were expecting. So things are starting to get a little more complicated. Next. Okay, now we're looking at diatom concentrations. And this was kind of interesting, the diatoms. So these are favored algae that we all seem to like. They decreased a little bit in the North Fork McCullamy River. You can see the warm colors on September 10th and then cooler in that middle channel on the 11th and 12th. Not much change in the other channels. So maybe a little response, maybe just something random going on. Next. Okay. And then looking at blue-green algae, so the ones we don't love as much. Sorry, Dylan, they just don't get as much love. But no, I shouldn't joke. It's a serious matter. But so for these guys, there was a slight decrease. You can see these warmer colors on the first day and getting a little bit cooler. So that's really interesting. And it's also going to be interesting to see what we see in the grab samples and see whether that matches up. Next. All right, so now we're moving into some of the grab samples that were taken off the boats and a couple of different forms of nitrogen. Up top here is dissolved nitrogen, on the bottom is ammonium. And the bars are set up in sets of 3. So on the left is Georgiana Slough, North Fork McCullamy, South Fork McCullamy River. And the colors are coded to the different days. So the orange is the first day where you had wastewater present. And then the gray and the yellow are for the 2 subsequent days when the wastewater from the treatment plant was not there. And there were significant decreases between that first day and the next 2 days in the case of dissolved nitrogen and ammonium, and also for the other forms of nitrogen, but I'm not going to show those here. So again, the grab samples are confirming what we saw with the high resolution mapping, that we did get that nutrient treatment that we were wanting to do for the experiment. And we think that's wastewater related. I'm supposed to say that, so I made it pop up. Okay, thank you. Next, please. All right, so now we're looking at turbidity, and this is interesting. We were not really expecting to cause some huge change in turbidity with the treatment plant going out of offline there. But light turbidity decreased, so less particulate matter in the water, so light increased. And that happened, and it was a significant change, and it happened to line up with what was happening with the wastewater treatment. But thankfully, the USGS has a high frequency sonde running at Freeport upstream of where the treatment plant discharges. I'm not going to show the data here, but we can see from the data from that sonde that the turbidity in the Sacramento River in general decreased, just lining up perfectly with our experiment. So we've got the situation now where we have decreased nutrients for the phytoplankton, but increased light. Next slide. And that's environment related. We're not saying that's the treatment plant. It's just something, maybe it's not the environment, something not treatment plant. All right, so now we're going to look at some of the phytoplankton responses. And again, things get a little confusing. And so on the top left here is chlorophyll, the same setup with the 3 channels, and then the days color coded. And for chlorophyll, there was not a statistically significant difference. Same thing with total phytoplankton biovolume. There was not a significant difference, but in the case of phytoplankton density, there was a significant across the days. And in the case of cyanobacteria density, there was a decrease across the 3 days. There's also some stuff going on with channels, which I won't get into, but usually the South Fork McCullamy pops out as being a little bit different if a channel is going to be different. And so there was a real mix of whether you saw a response if you're looking at the pigment, whether you're looking at the volume, whether you're looking at cell counts. So just a lot to delve into there. Okay, and so maybe this is wastewater related. It certainly lines up. And again, this decrease here might be wastewater related, but we're not sure. All right, so looking at phytoplankton productivity, this is where things got really neat because of that turbidity change. Phytoplankton productivity actually increased. So if we're seeing any trends with the phytoplankton abundance, it was either the same or a decrease. But in the case of the productivity, it actually increased. And so if you look at the left-hand graph, it's carbon uptake. And it's actually showing the Sacramento River on the far left and McCullamy River on the right. The three middle panels are the same three side channels that we were just looking at for the other graph. And the same thing with the dates. You'll see that the orange line is there, and then the gray line's a little lower, and the yellow line is a little bit higher. And according to Monet, what happens is if you blast phytoplankton with more light than they're used to, they don't do as much uptake for a day, and then they go, hey, I like this light, I'm acclimated to it now, and I'm going to increase my productivity. So she said that's what you would expect if there was light coming in and the phytoplankton are happy with the light. In the case of the right-hand panel here, it's Delta 13C enrichment of the phytoplankton, the particular organic carbon in the system, to be more specific. And as that gets more negative, it's a sign of increased productivity. So what you're getting here is increased productivity across the days, and also interestingly, increased carbon uptake and increased enrichment as you go from west to east. So those channels actually get shallower as you go from west to east, and longer retention time in the South Fork. And what do you know? You're getting increased productivity over there. So the phytoplankton are actually kind of doing what you'd expect in terms of light. And so probably environmentally related, because if anything, if you take away nutrients, you think you're going to have less phytoplankton, but we're seeing a trend here in productivity that would probably eventually have led you to increased biomass. All right, so zooplankton, this was kind of interesting. This is the total zooplankton density. And this also was true for the Cladocera, which is like Daphnia, which is a happy phytoplankton that people love. This one, statistically, it showed that there was a decrease by day, which we weren't expecting to see. But if you look at the graph here and look at the statistics, it's driven basically completely by something happening in the South Fork McCullamy River that we don't really know what it was. The trends in the other two channels were flat. If there was something going on with nutrients and those changed in all the channels, you would expect to see it happen everywhere. So click one more time. There's kind of a lack of consistent change, I guess I would say, with the zooplankton, and that's what we would have expected. But again, interesting things happening over in the South Fork. And just so I'm going to stop saying that about the South Fork, a lot of people in the research group really got excited about this. And I think a subsequent follow-up, and it does tie into Delta Nutrient Research Plan objectives, is to go and look at some of these peripheral, shallower areas in the Delta and look at productivity there. Because there's something going on in these shallow areas that you just don't get to see if you're out in the main stem Sacramento. And Brian is nodding, and you can be the one who goes and does that study. Next, please. Okay, so for zooplankton growth, just going to focus on this bottom panel for the growth rate. It fluctuated by the day, but no clear increase over time. So sadly, the zooplankton did not appear to be gearing up and showing increased growth as we decrease the nutrients. Next. And again, that's what we expected, not a big surprise. All right, and clam biomass, again, we were just looking at this to get a sense of what might be happening with grazing. So you've got biomass on the left and clam turnover on the right, and they correlate together very well. On average, the clams were able to graze about 2% of the biomass per day, but a maximum of 8%. If you look over at one of the South Fork McCullamy stations, this is the first time that we know that anybody actually sampled clams in these channels. There were a lot of clams, almost 24,000 clams had to be measured because you can't subsample them. So yay to Tim and the interns who got that done. Yeah, you get jars and jars, and you have, we talked to Jan Thompson, who's the wizard on this, and she's like, no. There's no way to subsample the way you would a phytoplankton sample, stir it up and pour off cart. No, you got to count the jar. So, okay, thanks. All right, so kind of wrapping this up then, I think I'm pretty close to time. So for going back to our food web, but here now on the left, we have the predicted for the side channels, not thinking about the Sacramento River anymore. Left is the predicted side channels, right is the observed. And so we were predicting that you might see a little bit of a decrease in the phytoplankton in that side channel. We did see a decrease in phytoplankton. If you're looking at just densities, and if you're looking at total density, if you're looking at cyanobacteria, not for the other stuff. So it's kind of like, I guess I would say if we're interested in HABs, you want to look into that. It sure would be cool if you could decrease harmful algae. But if nothing else is decreasing, then you have to kind of wonder, was it just a blip in the system? And so I think looking longer term now that the treatment plant is doing its biological nutrient removal, it's not as big a decrease in nutrients as we were able to do for this experiment, but it's more consistent over time, and you can look long enough that you're not gonna, you can kind of average out over things like that turbidity change that we saw. Next. Okay, so we predicted they'd see this ammonium decrease. Next. And we got that decrease in ammonium. Next. We predicted that we'd see a decrease in phytoplankton. Next. And we did see the sort of mixed signal. We didn't see a change in chlorophyll, but we saw a decrease in some of the density counts. But then we also saw increased productivity, which is kind of interesting. Like if you think you're limiting the phytoplankton by cutting out the nutrients, but now you give them more light and they grow, I don't think they were limited. So, but that's a Monet question, not a question for me. Next. The zooplankton and clams largely unchanged, at least across when you look all the different channels. And so one more, I think I'm done. Yeah. Okay. So now going back to our pretty picture of the conceptual model and what we actually saw. Next. Okay. So we were able to get that treatment from the treatment plant turning itself off. Next. And we did see the decrease in ammonium and lower levels of nitrate downstream. Next. And we did find out from the modeling from Resource Management Associates, the flow modeling, that we did have slower flows in these side channels. We did see increased light. We did confirm that they're, shallower than Sacramento and shallower as you move from west to east. This is done based on the density counts. So this is where you could argue about whether or not we actually saw decreased phytoplankton. This is the big, if you want to measure chlorophyll, you'd say no. If you're looking at density of cyanobacteria, you say yes, we did see a decrease. So I want to point out this could have a giant question mark beside it too. I just didn't put one in there. Okay, next. For the zooplankton, no change that we could see related to the treatment, and no change in abundance, and also no change in growth. Next. And the clams, we're assuming they didn't change. We only measured them once just to get an idea of what they were doing, but there wasn't, unless there had been some gigantic flow event that flushed them out like you would have seen in the wintertime, no reason to think that abundance changed over 3 days in October. Okay, so to sum up then, we did get that nutrient removal that we wanted to see in the river. We saw statistically significant declines in cyanobacteria and total phytoplankton density, but not in any of the biovolume measurements, and not in chlorophyll A. And productivity increased, but it seemed to be more related to that change in turbidity than to changes in the nutrient loading. So now I have my wall of text because everyone seems to get one today. And it's mostly, this is just so the steering committee has it to take away. But I looked through all the data gaps in the Delta Nutrient Research Plan and thought, which ones did we even touch on? I'm not saying we solved any of these problems. It's kind of like what Dylan said. Everyone seems to sort of touch on them. But we were able to look at, when you think about what conditions do cyanobacteria blooms occur, I thought it was interesting. We didn't have cyanobacteria blooms before or after we were out there with or without nutrients. We were able to look at a number of different factors affecting phytoplankton abundance and growth, although over a short period. So good data there if you want to plug stuff into a model. We've got lots of data for that. Looking at previous light and nutrient conditions, it was neat that we had the light and the nutrients, and we're able to see how those interplayed. Harmful algal toxins, we did check for that, and I didn't go into it today, but in terms of the cyanobacteria species, they were predominantly ones that are not going to be causing you trouble the way that microcystis does, so that was interesting. We did get over into some of the peripheral habitats, sampling in the South Fork McCollum and near those side sloughs. And then we have information on grazers, which would also be interesting for modelers to plug in terms of grazing effects on phytoplankton. All right, and then for future research projects, as I mentioned, I think it's going to be interesting to see what happens with the projects that are currently out there studying what's happening in the river now that the treatment plant is removing nutrients, much more nutrients than it did before. And then my recommendation, and it's been said, I think, by just about every speaker today, is make sure you're looking at all the factors. When you go out, it costs more money, but you're going to hopefully have better things to tell your managers in terms of how management actions are going to play out. I think there's just one more with all the acknowledgements. And I have like 2 minutes. No, I think I had 35 minutes. So.
Yeah, no, you're fine. So you can have a bathroom break or you can.
No, nobody leave.
Are there questions for Lisa? First in the room.
I know some of you have seen this before. The statistics have been redone courtesy of USGS internal reviewers, but the take-home message has stayed the same thing. Is there a question? No, just a wave. Oh, hi.
Hi, that's really interesting study. Did you guys model or like talk with the name more about like how looking at the growth rates, like how much chlorophyll you would have expected to see increased? given
Oh,
that-
based on that.
Yeah, based off of those
Yeah.
like, rates that were there and like. the
We
knowledge of,
didn't
like,
get
the
to that
residence time and how
point.
long- okay.
Yeah, it's just such a slew. The trouble with collecting data from that much of the food web is you end up kind of buried in data. You kind of, I could think if I just measured fewer things, it would be so much shorter. Yeah. Yeah, I'm not sure that'd be interesting. It's kind of more of a modeling calculation. I'm sure she could do it because she's done stuff like that for other projects where you predict as you move down the river with uptake rates at one station in the river, how much chlorophyll would you expect at the next one? Actually, I should mention, we just got a paper accepted, Tim just got a paper accepted in San Francisco Estuary Watershed Sciences, looking at the main stem Sacramento, but starting upstream, way upstream of Sacramento around Knight's Landing and working all the way down to Isleton, where Minay built a mass balance model where we look at what chlorophyll do you predict as you move down the river and what chlorophyll do you see, and it's taking into account the uptake rates and the grazing rates by zooplankton and plants.
Yeah, are there any questions on the line?
I don't see any
I
hands.
don't see any.
Hey, this is Vamsi Sridharan again. For some reason, my hand raise thing is not working. So great talk. Question I have is, did you guys look at the tidal exchange between the three sloughs during the time when you performed the experiment to see what sort of contributions you were getting in terms of the mass balance from potentially one of the other sloughs on? So when I say slough, I mean like the
The little
South
ones on
Fork.
the side.
Yeah, the Georgiana and the two forks of the South Fork and Colony. So did you see any exchange between the three?
That is a great question. We assume that it happened, but we don't have modeling data for there. And it was one of the most sort of hair pulling out parts of the exercise for for Marianne Guerin and Richard Raschiel, who did that work for RMA. Because we also didn't have good calibration gauges in the side channels themselves because they blew out in 2017 and had the USGS flow gauges and they hadn't been replaced. By the way, Vamsi, I really like the work you're doing on the winter run Chinook, and I see you in those meetings, so I'm just sending a shout out to you. I'm staring at the computer where your voice is coming from, and I probably should look at you here. That would be better. Yeah, so we assumed that it was happening. RMA and their report write-up said, you know, there's got to be stuff happening, but we weren't able to quantify it. But you can kind of see if you look at the data, when you look at it sample by sample, you can see, oh, conductivity goes way down. Well, probably that's water from the McCullamy River, and then conductivity goes way up. It's kind of Sacramento or from one of those little side sloughs. And it gets broken up. So the other thing that RMA was modeling was how the tidal flux in the three side channels was affecting the water. And if the tide was reversing, the water in the McCullamy River would actually get pushed upstream, and you'd have Sacramento River water in the McCullamy, and then as the tide went up, it would suck water back out. Yeah, so there's definitely a lot going on there, but without better gauging, you know, we were lucky we were able to model it. I didn't want to go out there and try and say anything about nutrients from the treatment plant without really knowing where the water was coming from to some degree. But yeah, there's a lot more complexity there. If someone wanted to go focus on the South Fork, you would definitely want to do more there. I should mention, though, that we did do cross-river transects at all the river junctions, but not those side sloughs. But we went out and did conductivity, temperature, DO transects at all the different channels each day at the end of the day to help them calibrate.
I just wanted to clarify that in Georgiana Slough and in the North Fork McCollum, the model was quite accurate per tidal cycle to tell us how the water from both the Sacramento and the McCollum were blending together and moving down through that part of the system. So the South Fork is a little bit harder to discern, but there is a full set of models for the other two channels.
And just one other thing, Janis, because this is important for people who want to go try this at home. We use the RMA modeling from previous studies to know how far downstream in each of the side channels we could go before we were going to get to the point where, because of tidal fluxes, we were going to be having water from the San Joaquin mixing in. Because we were trying to just look at what's happening with Sacramento River water and the wastewater treatment plant. And if we'd gone any further downstream, we would have had nutrients from the San Joaquin mixing in and really confounding things. So there was a limit to how far away from the treatment plant we could even go with a study because of not being able to isolate treatment plant water. And I'm gonna, the cane is coming.
That's a really good point. Thank you so much, Lisa, and thank you for the shout out too.
Situation definitely handled in the study. So next up we have Tamara, Kraus, and Keith. I think you're tag teaming this, right? So again, on the theme of holistic monitoring of phytoplankton, other water quality parameters, chlorophyll, and harmful algal blooms. So from our Water Science Center at USGS, Tamara and Keith. Thank you.
Okay, thanks for the opportunity. I think I have a lot of ground to cover here, but a lot of there's been a lot of allusions and references to the work the USGS does, which sets this talk up nicely. So our group really does focus a lot on looking at nutrients, not just nitrogen and phosphorus, but we're also interested in carbon, dissolved organic carbon. We're not going to talk about that today, and phosphorus. And starting to develop those really critical linkages to phytoplankton. And I want to make a call out today. We're going to focus on harmful algae, but we do really not, we don't want to forget about beneficial algae, which has also been mentioned, that we're not only concerned about nutrient effects on the bad kind of phytoplankton, but we really want to promote the beneficial phytoplankton that promote the food web. So I'm going to talk kind of more generally about the work that we've done. And then Keith is going to talk a little bit more specifically on recent data we've collected related to cyanotoxins. And later you'll hear again about the work our group does from Brian as well. And Joe Dalmagowski, who also is at the USGS, is also giving a talk later today. So next slide. Again, I wasn't sure about the audience, so I just want to give a quick big picture and remind people about how the USGS California Water Science Center operates. Our mission really is to collect data to address water issues facing the state of California. We do that by writing proposals, and the only way we can get our proposals funded is really by collaborating very closely with cooperators like the Delta RMP to understand what the main issues are. So we do really look a lot at the Delta Science Action Agenda, as well as the Nutrient Research Plan, to try and understand what the whole community thinks is important to study and try and meet those needs. And we do that, again, by collaborating with lots of agencies. And I want to point out that a lot of these agencies collaborate with us as colleagues and not just as funders. But that's really great to work in this system. Just something about our group is we're now almost over 40 people. We own a lot of in situ instruments. We do process a lot of samples. We've got sensors out at stations and fixed stations, either on pilings or in buoys. And then we also do a lot of this high resolution mapping I'll tell you a little bit about. Next slide. So we study a range of topics, but one thing on the right here, just a little conceptual model of, I think, what gives a lot of the work that's done in the Delta, a lot of sort of leveraging all the different efforts, is there's a lot of high resolution monitoring at fixed stations, so places that USGS, also DWR, goes out to continuously and collects data at specific locations. And that gives us a lot of information about what's happening over a very important temporal scale and changes over time. But we've also recognized that we need to get to more locations. And to solve that problem, we put our gizmologists, Brian Bergamaschi and Brian Downing, who used to work in our group, and they figured out a way to get all those instruments onto a boat, flow water through it. And we like to test new sensors and put them out on the station as well as out on our boat to try and collect more data. Because our philosophy is that because the Delta is such a complex place, we really need to try and collect data at both the relevant spatial and temporal frequencies that are needed to really understand the system, and then relate that to all these drivers in this really complex system. So lots of studies focus on just understanding concentration, sources, and loads in the system, as well as with phytoplankton, you think of it as abundance and composition. But then understanding what are the drivers that everyone's been talking about, that there are these multiple drivers. I think Brian's going to talk about the bottom bullet points here about how we're always looking at new tools and approaches. We're also interested now with phytoplankton getting into relating the data we collect to remote sensing, and then improving data access, integration, and visualization to help improve our understanding of the system. Next slide. Okay, so we've had a couple of different conceptual models. This is just another conceptual model saying the same thing. It's complicated. So we're biogeochemists in our group, so we kind of like to get into the weeds. When we picture a parcel of water moving through the delta, we kind of imagine that every little cell that the parcel is passing through in the delta, all kinds of things can happen. If it's cold out, maybe not so much happens in the winter and things flow through really fast, but definitely in the summer and areas where water's sticking around, there's all kinds of stuff that can happen to your parcel of water. And so, if you think of it as just a box, you think there's inputs and outputs, and then you can kind of model what's happening inside. But really, you have to break it down and try and figure out what are the most important processes that are acting on your parcel of water. And in this case, since we're talking about nutrients come in and then they react in that water parcel that you're thinking about, that cell, some location. And we're really interested in particular how nutrients are interacting with phytoplankton, both the harmful kind and the beneficial kind, as well as aquatic macrophytes. And I want to point out that it's like a two-way street. Nutrients affect the biology, but the biology also affects nutrients. And then shout out again, it's complicated. It's not just nutrients that, it's the nutrients come in, depends where you are, depends how long you're taking to pass through the system. There's environmental complexity, hydrologic complexity, and landscape scale complexity, water depth, light, temperature, all those factors. So this is just the conceptual model that we all, I think, have in our mind about why it takes a lot of studies to understand what's going on in the system. Next slide. So Tim covered this pretty well, but I just wanted to give a shout out to the work that we've done that's specifically been funded by the Delta RMP. We started pretty much early on. We were asked by the Delta RMP to work on these 3 reports that came out. One was kind of an introduction on like what are high frequency measurements, and we put some case studies in at that point to how we found that what kind of information we were getting out of collecting data at both higher temporal and spatial resolution than just monthly grab samples. There was some data synthesis in there, looking at the data that we had to date. And then we were also asked to literally put some dollar amounts on what would it cost to put a high frequency monitoring, a nutrient monitoring program for the Delta. What would that look like based on existing technology in 2016? The Delta RMP did a great thing for our group, and I think the Delta by funding the first Delta-wide mapping campaign. So we'd already developed this ability to put all our instruments on a boat and drive around at high speeds and collect high resolution data, but we hadn't really taken it to like, let's do it across the entire delta over a 3 to 4 day period. And in 2018, we got funding from the Delta RMP. We didn't really understand at that time what we were committing to. But somehow we figured it out, and now we seem to keep doing it. And we generate these kinds of maps. I'll talk a little bit more about that. And then Keith is going to talk about the cyanotoxin monitoring that the Delta RMP has funded. That's allowed both the USGS and DWR to collect samples and get them analyzed for cyanotoxins, which are quite expensive to analyze for, which is why we don't have a lot of data on cyanotoxins, because, you know, if you're out there already collecting samples for lots of other studies and you grab a water sample for cyanotoxins, that part of it can be a sort of a cheap leverage, but then actually analyzing the data at the laboratory and then analyzing it to figure out what it means needs some extra funds. We participate in the chlorophyll intercal study. You saw we participated in the shrink study Lisa just presented, and we've hopefully Dave will talk about how he uses some of our data, and generally too when they talk about modeling, as well as data that DWR generates. Okay, next slide. This is also my slide for people to look at later. Those are the 26 questions that we were asked to talk about how our study is related to. Blue are kind of the main nutrient ones, so 8, 16, and 17 I think is what we mostly hit on. Green are more phytoplankton stuff, and Keith can talk about how we've addressed those. But the focus has really been on making sure that we do have the data to understand status and trends, nutrient concentrations, their forms, the ratios of nitrate to ammonium and to P, collecting data on phytoplankton, not just chlorophyll concentration, but species composition, and starting a program that's really looking at cyanotoxin concentrations across the delta. And then the goal is, and I think where the power of like leveraging on the work we're already doing is we're already out there collecting the basic water quality parameters, temperature, pH, salinity, DO. Most of our stations are co-located with our hydrodynamic group's flow monitoring network, so we can look at fluxes past those fixed stations, and then relating nutrient concentrations to what we're really interested in, which is the phytoplankton and the macrophytes. Okay, next slide. I threw this up here too, because I don't, just another shout out. I think we're doing a good job as a community at hitting the original assessment questions that the Delta RMP had set out to answer related to status and trends, sources and pathways, processes, and forecasting. So that was just another, I just wanted to remind us all that we have these assessment questions too. Next slide. Okay, so I want to spend a little bit of time on this because for those of you who aren't familiar with our particular program, again, on the left here, I'm showing where we have our fixed monitoring stations in the Delta. There are a lot more stations in the Delta besides this. So on the next slide, I'll kind of highlight why these stations are more important maybe for the nutrient monitoring group. But remember, DWR has lots of stations out there that measure flow, and a lot of those also measure some of the basic water quality parameters. But these are the stations that our group runs. And just as a shout out there, we just put in 3 new stations here that are actually on the island drains. So we're trying to also now get at nutrient fluxes from island drains. We'll see how that works. Kind of some dirty, turbid water to put in situ sensors in, but we're trying. And then this here is just to point out that when we do our boat-based mapping campaigns, that's the coverage that we generally have of the Delta-wide mappings. We also do like more targeted studies that look at smaller portions of the Delta where we can throw in additional instruments or maybe go out for, to try and answer more specific targeted study questions. And then those black dots are where we stop and collect data. So again, temporally dense data collecting on the fixed monitoring stations. We're collecting data every 15 minutes. Discrete samples approximately monthly, whereas this mapping right now, we've traditionally done that spring, summer, and fall, so 3 times a year that we've generated those maps. Next slide. So again, since these will be available later, I just wanted, for people that aren't familiar, this is generally the instrumentation we have out at our fixed stations. We have a fully loaded YSI. What we put out there that's potentially different than a lot of other groups in the Delta is that we also have fluorescence of dissolved organic matter in addition to fluorescence of chlorophyll fluorescence. And we also have SUNA nitrate analyzers. So that's where it really ties into the Delta nutrient monitoring goals, especially more relevant now that there's less ammonium in there, because before we only got ammonium when we went out and grabbed a sample. But we did, under the funding we got for all our different mapping campaigns, we do have a flow through ammonium analyzer. So we put that analyzer on the boat. We still have like dreams of putting ammonium analyzers out at fixed stations, but that has yet to be realized. And now we don't actually see that much ammonium in a lot of the Delta, as you'll see, not the wastewater treatment, but upgrade. So, and then discrete samples. Keith will talk about how, in addition to a lot of the basic parameters, we're now collecting cyanotoxins at a subset of stations. Next slide. Okay, this is just a figure to show the kind of data that we produce. A lot of squiggly lines at the fixed stations. You can see there's sort of the seasonal beat. This is 3 years from about 15 stations. You can see that there's quite a pattern in temperature, obviously, but when you squint at it, there's not always a seasonal pattern in a lot of other parameters. So that's why it'd be interesting to understand what those patterns are. And orange here is nitrate. But that's the kind of data that we hope modelers will find extremely appealing to imbibe into their models. Next slide. And then this is the kind of data that we are generating from our boat-based mapping campaigns. Again, a lot of information that we can glean out of these. And what I want to just highlight in the next couple of slides is that we can look at this data and we can query the data for status and trends, and we can start looking at relationships between parameters. We can try, and now that we have so much information, we think we're getting the kind of information we need to understand environmental drivers, temperature, drought events, that we're collecting data over time. And then also start looking at impacts of management actions like the wastewater treatment upgrade, barriers, flow actions, things like that. Next slide. So again, just reminding you that there's complexity to the system, and I'll just talk about two really important factors. Next slide. I think Tim touched upon this. Hydrologic context. So hydrology really matters in the Delta. The first way to look at that is water year type, but there's also the timing of precipitation, how much snowmelt we have, reservoir releases, barriers affect where water flows, and gate operations like the Delta Cross Channel can really affect where like Sacramento River water ends up. So there's a lot of complexity to the hydrology, and that will impact water quality. It'll impact water residence time, which is of course a major factor. The more time water is going through the system, the more these biogeochemical forcings can transform nutrients, and it affects mixing and transport. And I just to highlight, in 2018, 2020, 2021, 22, those are the years we've done our broad area Delta mapping, our fixed stations, and DWR EMP has been going for years collecting data. So they have data across water years, but really the last couple of years, we have only really been collecting data during low flow conditions. So we just need to keep that in mind as we interpret our data, that we aren't collecting data that we can extrapolate into much wetter hydrologic conditions. Next slide. I wasn't sure whether anyone would talk about the wastewater treatment plant upgrade, so I'm just going to highlight that really quickly. That's pretty much been a big change in that anyone studying nutrients has to think about when we're studying the system. The idea is that by the wastewater treatment upgrade, by doing nitrification and then denitrification, not only is the wastewater treatment plant now putting out not putting out ammonium, they're putting out nitrate. They've also reduced the total amount of nitrogen that's entering the system. Next slide. And this is actually data from regional sand that actually shows their effluent nutrient concentrations. So this is what's in the effluent, not what gets diluted in the river, but I wanted just to put it in context for anyone interested in our mapping data. This is 2018 when we did our first set of mapping data. That was well before any wastewater treatment plant operations were started. You can see the nitrification, the side stream nitrification started, bumped up nitrate a little bit. The orange line is nitrate. The blue line is ammonium, and then yellow is the sum of dissolved inorganic nitrogen. So puts it in context that, you know, in 2022, we're going to go out next month. We are now well beyond the wastewater treatment plant. So I think we really can start asking the question, and we are very excited to start querying the data to see what were the impacts on nutrients, but also phytoplankton abundance and community composition. Next slide. This is just to show, okay, we can look at our fixed stations. This is Walnut Grove downstream of the wastewater treatment plant. Again, orange is nitrate, sorry, at our high frequency nitrate sensors. And yeah, you can see nitrate concentrations are now higher in the river. Next slide. And then now we really have quite a lot of work to do to figure out how we're going to analyze this geospatial temporally dense data that we have for our mapping campaigns. Just wanted to quickly point out, even looking visually at that pre-wastewater treatment plant upgrade, high ammonium in the Northern Delta. Now in 2022, this is spring. The top line is ammonium. You can see there's a decline in ammonium. If you look closely in the North Delta, you can see there's an increase in nitrate, but if you're concerned more about the sum of dissolved inorganic nitrogen, nitrate, and ammonium, then that's when we start having to look more carefully to see if there really is a significant change. And then the next question is to put up all the phytoplankton data, look at abundance, but then also see, are we decreasing? Do we see a change in our harmful algae? And do we see a change in our beneficial algae? Next slide. Okay, how am I doing for time? I have three more points I just want to quickly make.
That's really between you two.
Okay, so I just wanted to highlight kind of three interesting things that I think we've learned from the studies we've done. One is kind of the residence time really matters. I think we all knew that, but the residence time really, you know, the more time the water parcel is in the Delta, the more nutrients are going to be transformed. In this case, in the Northern Delta, we see that nutrient nitrate is coming up. This is nitrate concentration. Red is high concentrations, 15 micromolar, all the way down to 0 micromolar. And you can see that as water is pulsing up and down with the tides into the Northern Delta, we really are using up all the nutrients. And so with the wastewater treatment plant upgrade, you would think that all those nutrients, there's going to be less of them. And is that going to affect phytoplankton production, aquatic vegetation in that area? We're really interested to see. And then you see the gradients are quite steep. And so you can see that when you drive around on a boat and map the data over probably a 1.5 hour period there that's covering. But then that's why you see these very big changes at a fixed station. So this is what nitrate looks like for this time period here in 2020 that you see if you just are sitting at that one station at Liberty Island, you really see the nitrate concentrations are high when you're getting water coming from the south, which has got higher nutrient concentrations. And then during ebb tide, when you start seeing water coming from the north, from higher in the system coming out, that's why suddenly you see a parcel of water that looks so different. And that's why these fixed stations, actually, when you think about them, they are showing you spatial variability because the water is transporting material back and forth. And that's why we need really smart modelers that can figure out what that all means, because because that's complicated to look at for my brain. Next slide. This is a graphic that we made just to show how rapidly we can see a drawdown of nutrients in the presence of high chlorophyll. So this is just a boat-based mapping campaign we did in Sassoon and Grizzly Bay once. And if you map, you see that there's much higher chlorophyll in these sort of backwater, more shallow areas. And when you look at the signal, when you see this high peak in chlorophyll, you also see the sudden decrease in nitrate. And that's something we see a lot is when we do our boat based mapping, or even at our fixed stations, that we see that chlorophyll definitely consume nutrients. I mean, phytoplankton do. And that kind of does suggest that if you do lower the nutrient concentrations at some point, they might run out of nutrients. And so that's what we're kind of looking for, to see if that would be good for harmful algae, but it might not be so good for our beneficial algae to run out of nutrients. Next slide. I think Brian might talk about this, but this is another study. There's another instrument that we like to throw on that looks at water residence time. So I'll let him talk about this. He's nodding his head. He's got this slide in his talk, I noticed too. So again, just showing that it's complicated. We know that nitrate disappears, but it seems to disappear at different rates in different systems. And so we'd like to understand why, so we can help us with our forecasting models. Okay, I think last slide. This is sort of, I think keep on this again, but what do we recommend going forward? I kind of sort of more of the same. We can't really let up on putting our efforts into monitoring the system and keeping track of status and trends of nutrients and phytoplankton and cyanotoxins. So we need to keep that effort going. But at the same time, we really do need to understand drivers and interactions in order to understand how the system works, so we can answer that question of are nutrients a problem, and if we want to change our nutrient inputs to the system or how the system is managed, what kind of effects do we think we'll see? And with that, I'm going to pass on to Keith.
Great, perfect. You can go to the next slide. Great. Yeah, so my name is Keith Bouma-Gregson, and I work with Tamara and Brian at USGS, and I'm rather new to the group. I joined last year, and prior to that, I was working for the state board, leading the freshwater harmful algal bloom program for that agency. So I worked with Janis and Meredith a little bit in that capacity, and so it's nice to be back at the Central Valley Regional Board. And my background's in algal ecology and cyanobacteria, and so I'm going to be specifically talking about some of the cyanobacterial projects that we've worked on. And also want to mention that a lot of this work was done in collaboration with Department of Water Resources. So I've listed some of the collaborators with that work, including Ted Flynn, who's here in the room with us, and that the funding, as well as has been mentioned, some of it has come from Delta Regional Monitoring Program. So you can go to the next slide. So cyanobacteria, just a little background, are photosynthetic bacteria, and they evolved over 2 billion years ago on Earth, and over the course of that time have changed a lot of the biogeochemical cycles on the planet, and also evolved to inhabit a lot of different environments on Earth. And they're primarily aquatic, and so they're found in almost all the freshwater and salty water habitats on Earth. But in freshwater habits in particular, when certain conditions are generated, they can bloom up and increase their biomass rapidly and start to create nuisance and harmful blooms. And so 3 of the types of organisms that you'll hear me talking about today are Microcystis, Aphanizomenon, and Dolichospermum, which all occur in the Delta. And when they bloom, you can get taste and odor compound issues and filter clogging and infrastructure issues regarding to drinking water intakes. They can degrade the aesthetics and recreational opportunities and experience of a water body. As the blooms start to crash, the decomposition of the bloom material can lead to low dissolved oxygen concentrations. Cyanobacteria are generally considered less palatable and nutritious than other algae-like diatoms, so you can have negative food web impacts, and also they produce toxic compounds which are harmful to animals. You can go to the next slide. And so, so these toxins, so they produce dozens of different types of molecules which are harmful to pets, animals, livestock, and these are generally considered secondary metabolites which means the molecules themselves are not essential for the functioning of the cell, which means that you can have some strains or species of cyanobacteria that produce toxins and others that do not, which adds a layer of complexity to understanding the ecology of toxin production, and then also even just understanding the impacts that a bloom might have when you start to see biomass increasing. Generally, though, there's about 4 different types of effects that the toxins cause, so they can affect the skin, kidneys, liver, or the nervous system. And in this table here, I've listed out some of the toxins that our group has been monitoring. And a microcystin at the top there has been the most frequently detected and well-studied toxin in the Delta. And really across the globe, that's the one that we know the most about. Some of these other toxins have been detected in the Delta, but at lower concentrations than microcystin. But in other systems, they can occur or they can be the dominant toxin. So we're kind of approaching these projects to collect data on a suite of toxins to make sure we have a pretty comprehensive understanding of which toxins are present in the system and where they might be occurring. And the last point I want to make here is that these molecules are quite chemically different and distinct from one another. So the genes that are used to synthesize them by the organisms and the roles that they play within the cells are diverse. And so we can't really extrapolate from toxins, because we really have to understand each class of toxins as like its own distinct entity. Next slide. And so a brief overview of some cyanobacteria research and observations in the Delta. In 1913, Winfred Emery Allen, who's an oceanographer at Scripps in San Diego, came up to the Delta and did a bunch of surveys on the San Joaquin River around Stockton. And these images here in the top right are from his manuscript. So if you ever wondered what San Joaquin and Stockton looked like in 1913, you should check out some higher resolution images from that. It's pretty cool. And in those plankton net toes that he did see cyanobacteria. So we know that it was at least here in the system back in 1913, but he doesn't really write of like blooms per se. So then the first report of a bloom came in 1999. And it was microcystis was identified, and that's a figure from that first report in the IEP newsletter. And since that time, most research has been done on microcystis and the toxin microcystin. And then in 2016, sampling from DWR was first detected anatoxins and saxitoxins, which is this figure here from a paper by Peggy Lehman. And those green dotted lines are the toxin, the detections of anatoxins that occurred that year. And then what's also kind of been recognized more recently is that there's a number of places that seem to have recurrent high toxin concentrations in the Delta. And so this here is a figure that Peggy Lehman put together for an IEP workshop using data from the state and Central Valley Board. And the red circles show places where the toxin concentrations exceeded the highest danger level recreational threshold that was set to the water board's criteria. And where these are generally located is Stockton and Discovery Bay have recurrent blooms. But then also there's been a number of more enclosed marinas where there's been surface scums developed in those places as well. And those have very high toxin detections. And you'll see that this is in a bit of a contrast from the data that's or the concentrations that have been observed at like the EMP stations or the stations that we've been sampling at as well. And then lastly, my final note here, I didn't have time to squeeze this on, is that there's been a lot of genetic work done in the Delta over the last few years, and that's provided a lot of interesting information about the strain variations in the Delta. And Ellen Preece, who's going to be presenting after me, has been involved in some of this work. And so it's I've been tracking her work excitedly and looking forward to seeing more results coming out of that. A lot of that's been Ellen, and then Tim Otten and Ben Genetics, and Janis has been involved with that too. All right, next slide. So now we'll get on to some of the projects that we've been doing at the USGS. Next slide. So as Tamara mentioned, we have these 2 core data collection efforts, and with our efforts at EMP, there has been a lack of, a few times have toxins been co-collected with all these other parameters that are being collected. So we can go to the next slide. So in about 2020, we began to get some funding to add cytotoxin sampling to our, the suite of other nutrient and water quality parameters that we were collecting at stations. And so this map here shows the fixed stations where the black and blue dots are the monitoring stations by USGS and DWR. And then the yellow stars show the locations where cytotoxins and additional phytoplankton cell counts were were being collected. And 2 of these stations, Rough and Ready Island and Vernalis, which are EMP stations, were done in collaboration with DWR, as I mentioned. So Ted's group and some other DWR staff collect there, and then the other stations are USGS stations. We sample these about 18 times a year, so it's monthly in like the off season, and then starting in April, we begin to sample every 2 weeks when we expect to see primary productivity increasing. And additionally, Frank's Tract here was we got some funding from DWR to do some work there this summer in relation to the emergency drought barrier, which is put in West Falls River, and there were concerns and interest in the potential for that barrier to change the HABs dynamics in that water body. You can go to the next slide. And so in flowing systems like estuaries and rivers, one of the challenges with cyanotoxin monitoring is that cyanotoxins can sometimes be moving as pulses, as you might get releases of toxins or dense concentrations in particular parcels of water. And so discrete sampling can sometimes miss those pulses. And so that's kind of to Tamara's point earlier, why we put in situ high frequency sondes for like chlorophyll and other parameters, because we know that just going out every week or month to collect discrete samples doesn't always tell the whole story. But there are no real like in situ toxin detection methods. And so these SPAT samplers help us get at a more temporally integrative method to look at toxins. And so what SPAT are is it's an adsorption resin that you put into a little mesh pouch, as you can see there in the river or being held up here. And so any dissolved toxins in the water column will then absorb and stick onto that mesh, onto the resin, and then you can leave it out for several days, absorbing and concentrating toxins, and then extract those off in the lab and measure what was absorbed to the mesh. And so these wind up being very sensitive, and you can often get detections from SPAT in cases where your discrete water samples are giving you non-detects. However, they're considered semi-quantitative because it's really difficult to fully characterize that absorption and desorption kinetics very precisely. And so it's hard to necessarily transfer the units from a SPAT sampler into the units of like micrograms per liter or things that, especially those units tend to be the ones that objectives are set within. But nevertheless, they've been a very useful tool, and they complement discrete sampling quite well, especially when you're doing some of the initial characterizations of a water body, trying to understand what you might be missing by just only looking at discrete data or discrete sampling. Next slide. And so again, the Delta Nutrient Research Plan data gaps. And so a lot of this work is, here's kind of the key ones that I highlighted or I felt like were relevant, and it is getting to, boils down to the idea of adding toxins into the suite of other parameters being measured, because we're just out, like there's a lot of incident response data that's collected where citizens might call up and say, hey, there's a bloom here, and we go out and collect toxins, but usually we're not collecting the full nutrients and water quality conditions along with those data. So it's hard to then align everything and match it all together, or then vice versa. We have stations like EMP and USGS where we're collecting everything, but not the toxins. So we're trying to fill that gap so we can get the status and trends. And then with that, it can lead to more predictive models and more mechanistic understanding of what's going on. Next slide. Okay, and so now a little bit of the results from our samples that we've got so far. So this is looking at the discrete water sampling done, and this is a combination of our fixed stations as well as when we collect discrete samples on our large-scale mapping runs. So we detected 3 different types of toxins: aminopeptins, microcystins, and anatoxins. The aminopeptins, you'll see, were the most frequently detected and also at the highest concentration. And now this is a more recent type of molecule that's being studied by HABs researchers. And the full, they're known to be bioactive compounds. So when people do experiments, they can see that like organisms respond and react often negatively to these compounds, but the full impact on human health is not very well characterized yet. So it's a little hard to know exactly what all this means. But what's kind of aligns with other systems where these have been studied is they do find them to be relatively ubiquitous. So they'll often find aminopeptins at higher abundances than some of the other toxin concentrations, which is kind of like what we see here. In terms of the other concentrations, The antitoxins, the microcystins, were below the warning level, recreational level, which is the no swimming advisory, which would be 20 micrograms per liter for antitoxins or 6 for microcystins. So we were below those. But again, as I mentioned, we're missing those known hotspots where, as Ellen's probably going to talk about, Stockton had much higher concentrations this summer. In terms of where they're occurring, it's primarily, or in this maps here, the red dots are the detections of the toxins. We're mainly seeing them in the central and south delta, less in the north delta, and we haven't had any detections out in Sassoon. You go to the next slide. And so when we look at the SPAT results, these are primarily, I'm just showing from our fixed stations here now, but what you see is that we actually detected, we had more detections, and then we also detected more toxins. So we also had cylindrospermopsin and nodularin detections from the SPAT in addition to those other 3. And that's sort of what we expect. We usually find that the SPAT are more sensitive than the discrete water samples. And spatially, one of the things that jumped out to me was that our Liberty station was pretty much detected all of those toxins, whereas when we look at our discrete water samples, we usually get non-detects up at Liberty. When you go up that way, you do sometimes see colonies of microcystis, but it probably suggests that things are relatively low abundance. So we're not always picking up in the discrete samples, but there is the potential for them to increase in their abundance. And then the additional information that the SPAT gives us is the timing of toxin production. So this table here has the microcystin and the SPAT and the whole water, and then the SPAT and the antitoxin, SPAT, and whole water. And if it's green, it means we detected it in that month. And that's the number of detections and samples that detected it. And you can see there's a lot more months where we're detecting toxins from the SPAT than with the whole water. And one of the things that is also interesting here is the antitoxins. We have a lot more antitoxin detections in March and April than we do microcystins. And so we usually have thought of microcystins occurring after the water gets above 20 degrees Celsius. But in March and April, the water's a lot cooler. So we could have a situation where we've got an antitoxin producer that's maybe has higher biomass earlier in the spring, and then that's getting succeeded by our like microcystis and the microcystin producers. All right, next slide. Okay, and now a moment to also talk about Frank's Tract, because there were some interesting observations from there this summer. So there was blooms in Frank's Tract both in 2021 and 2022. And so this here is a histogram showing of satellite data. So basically the x-axis is time, the y-axis is the number of pixels that detected a bloom. And so you can see here in 2021 and 2022, we had these big bloom events, but nothing in 2020 on the satellite, up on the satellite. And so DWR asked us to go out and do some research on Frank's Tract this summer. And so when we went out there, one of the first things that stood out was that the bloom was not dominated by microcystis, where that's usually the organisms that we've seen in the delta. It was dominated by Delicospermum. And we also saw a fair bit of Phanozomenon. And one of the things that distinguishes these organisms from microcystis is that they can both fix nitrogen. Now, we don't actually know if they were fixing nitrogen, but they at least can fix nitrogen, but microcystis cannot. It also wound up not being very toxic. So even though the water was quite green and the abundances of cells was high, we had very few toxin detections, and it was basically just on the SPAT that we picked it up on. So if you go to the next slide. So one of the things that's got me thinking about is the relationship in the literature between microcystin and nitrogen. And so this here is an image of the microcystin molecule, and it has 10 nitrogen atoms within the molecule. So when cells synthesize microcystin, it's a pretty hefty nitrogen investment that they make. And research has shown that there's a decrease in microcystin production when nitrogen gets low as it decreases. So it could it's both a combination of, you can see a switch in the strains where non-toxic strains will begin to dominate the bloom and outcompete the toxic strains. And then the toxic strains themselves will also downregulate and decrease their production. And so some data from this, okay, thank you, is this here shows data from Western Lake Erie, and this is microcystin production up here and nitrate concentrations in the lake. And you can see as nitrate drops, the microcystin decreases. And then these are, this is some experimental results showing their initial and control. toxin concentrations, and when they add nitrogen, they get a lot more toxin production. You can go to the next slide. So there's a lot of nitrogen gradients here in the delta. We see some of the highest concentrations from our mapping data in the San Joaquin River channel around Stockton. This is in micromolar, where we're well over 100 micromolar. And then as you move westward, you see those concentrations decrease due to phytoplankton uptake, as well as dilution and dispersion with Sacramento River water that has lower nitrogen concentrations. And so after being out on Frank's Track this summer, observing the shift in community, it's got me wondering, is there a relationship between the abundance of microcystis and the amount of microcystin to lower inorganic nitrogen concentrations as we move around the delta? As we've seen, the highest concentrations often co-located, where we see the highest toxin concentrations can be co-located with the higher nitrogen concentrations. Next slide. And so to get back to our data gaps, just trying to fill in some of the progress we've made. So in terms of when and where are we seeing blooms occur, a lot of it is in the south and central delta. Though some of that SPAT data might be showing some potentially low abundances of toxin producers up in the Cache Slough complex. Also want to highlight too that some of these blooms were having impacts on the drinking water plants. Even though we didn't see toxins in Frank's Tract, there were some taste and odor issues, as well as some filter issues and clogging issues that were occurring in the southern delta. In terms of the range of toxins, the microcystins, as we know, but we're now starting to see different additional classes of toxins that are there. So it's sort of backing up that finding that DWR started to first observe in 2016 when they saw new toxins occurring. And then in terms of more of the predictive relationships and modeling, we're going to start working on some of our reports this winter here once we get all our summer 2022 data back. So stay tuned for more statistics and analyses. And one of the things we're definitely interested in is this relationship between nitrogen and microcystin production. Next slide. And in terms of future research projects, I think the more research on some of these additional producers that are out there, we really don't know who's producing anatoxin or who's producing cylindrospermopsin. Monitoring studies to compare what's going on in some of these hotspots, like Discovery Bay or Stockton, to the long-term stations. So we have a better sense on how to compare data there with like the long-term records, those areas, nitrogen microcystin production. And then also we could be great to learn more about the ecology of Delicospermum and Aphanizomenon, as we can see that that's what was causing the issue this summer in the central and south delta. Next slide. And yeah, so big shout out to all the field staff and all the different agencies that are out collecting these data, because we couldn't do it without you and those who funded us. So thank you.
Time for maybe a couple of questions. So any questions in the room for Tamara or Keith? Ted?
One of the science papers you're, I remember they talked about the role of microcystin in protecting from reactive oxygen.
Yeah.
Species. I don't know much about actual like measuring reactive oxygen species in the field and things like that. How practical would that be to look at that too, in terms of another environmental driver and production of those?
Yeah, I think a lot of that is going on intracellularly. So like kind of within the cell, like at the membranes where photosynthesis is occurring. So I don't know if like external, how far you get like in the environment, but we could look into that more.
Okay, well, Brian, we were gonna alternate, so Brian, anybody on the,
I don't see.
I don't see it in person.
okay, all right, go ahead.
Yeah, just curious, what satellite were you using? For
Oh, yeah, that was the Sentinel-3 Ultra sensor, which is about a.
Is that a hyperspec?
No, it's a multispectral, and it's what's used, kind of the main sensor used by NOAA. They developed the algorithm for cyanobacterial blooms using that sensor.
Yeah, so in 2021, if I remember right, one of the interesting findings we found was that nutrients weren't like depleted, they were still a lot of them around. Was that right? I was just wondering.
Or
The one in Frank's Tract in 2021. And so I was wondering, I didn't realize, Leucospermum and, the Anapina that they're nitrogen fixers, so I was wondering if. And I just find that like an interesting relationship to maybe highlight for the group of like.
Yeah, so we actually, we didn't do as much work in 2021, specific in Frank's tract. I think we were around it more, but definitely we haven't got a discrete data back, but nitrogen was much lower. Like when we were out on the boats with the flow through, you could watch, the gradient of like, as we left the San Joaquin, nitrate was decreasing in Frank's tract. Again, I'm not sure if they are fixing nitrogen. It's an energy intensive process. They usually need a lot of light, but with all the SAV in Frank's tract, there is less suspended sediments in there right now. And it's shallow, so there could be enough light for them to be fixing nitrogen, but that, yeah, we kind of need to actually confirm that for sure.
Yeah, I just remember like, how are there still nitrogen, or how is there still nutrients in
Yeah.
Frank's tract?
All right, thank you, Tim. Not least for this session, Preece from Robertson-Bryan has been involved in a number of HAB studies since 2020, some of which we've accepted funding from Delta RMP, so thanks for that.
Yeah, so I'm Ellen, and I've been working on harmful algal bloom toxins and harmful algal blooms in the Delta for the past 7 years. But my background goes beyond that, where I previously focused on lakes and such.
Those are my bonus slides.
Oh.
So, there we go. So Keith's talk was the perfect background and segue into my presentation. So thanks to Keith for that. But what I'm going to do today is talk about 3 studies that I'm involved in. They're ongoing, none of them are complete, and the relationship of project findings to the knowledge gaps in the Delta Nutrient Research Plan. So first, I'm going to give an introduction on these algal bloom projects. Each project could have a whole presentation devoted to it, so I'm just going to give a very high-level overview. I do want to say Central Valley Water Board is a partner in all 3 of these projects. I have numerous collaborators, but they are the constant collaborator on all the projects today. So I'm then going to present 1 to 2 slides on spatial findings. So what am I seeing across these studies, and what are we seeing across the Delta? What are some findings from that? Same thing with seasonal or temporal findings, 1 or 2 slides on each project, and some patterns that we may be seeing related to temporal things. And then I'm going to talk about project findings and the knowledge gaps. So I've chosen 3 knowledge gaps that I'm going to focus on and discuss how our project findings are starting to fill those knowledge gaps. And then finally, the same 3 knowledge gaps. What are some next steps? What can we do next with those 3 knowledge gaps? Next. Okay, so this is the first study that I'm going to discuss. I'm calling it the shellfish study. And this is identifying cyanobacteria harmful algal bloom toxins in Delta invertebrates. The goal of this study is to determine if microcystin and saxitoxin are stressors on food webs and native fish, including managed species. The focus here was really on sturgeon, and so we worked closely with CDFW and the DWR sturgeon team to identify 10 sites in the Delta where sturgeon may be feeding, and we tried to overlap that with known clam abundance and then as well as harmful algal bloom, known harmful algal bloom presence. We began sampling in July 2020, and this month is our final sampling date. And so we collected 17 sampling events per year. So similar to Keith, we really focused twice a week during the summer months and once a month during the winter months. Collected Asian clams from the sediments at these 10 sites on each event. And then when possible, we collected crayfish. It's been a little more difficult than we expected. We set the traps and just aren't having a lot of success. We have caught some. And then we're measuring microcystin and saxitoxin in both shellfish and water samples. This project is funded by Proposition 1, and partners are San Francisco Estuary Institute, Central Valley Water Board, Tim Otten at Ben Genetics, UC Santa Cruz, and then Fishery Foundation is collecting our samples. Next slide. So the second study is what I'm calling sediment study, and this is mapping benthic overwintering microcystis cells within the delta. So our goal is to determine if microcystis blooms throughout the delta are generated by benthic resting cells from a few select locations. So basically, microcystis, they bloom in the summer and then they settle out of the water column into the sediment, and then they can remain in the sediment over winter until conditions are good again for them and they can re-enter the water column from the sediments. This project is also a 2020 to 2022 sampling period. We have collected all of the samples for this project, but we're still in the process of analyzing them. So we collected sediment samples at 8 locations, the map on the left there. We collected samples in November, so the end of the blooming season. This is when we'd expect to see the greatest number of resting cells in the sediment. And then we also collected sediment samples in April. So this is after winter flushing, but prior to the cells entering the water column. And we wanted to see how many of those cells were flushed out from the winter rains and the higher flows. We expected that dead end sloughs would be the seed stock. Those would be the main producers holding the most benthic resting cells. And those with greater hydrologic connectivity would be flushed out with little remaining sediment stock in April. We used qPCR to quantify the microcystis seed stock in the sediments. This project was funded by Delta RMP, the Supplemental Environmental Project Funds, and also state HAB funds. And partners are Central Valley Water Board again, and Tim Otten with Ben Genetics. And then the final study that I'm going to cover today is what I'm calling the Stockton study. And this is us monitoring harmful algal blooms and water quality across the Stockton Deepwater Ship Channel, starting up kind of near Buckley Cove and then working our way down into the waterfront. So the goal of this project is to characterize how algal blooms and associated cyanotoxins change across time and space in the Stockton Deepwater Ship Channel. We're sampling at all 5 of the sites on 11 dates, and we're collecting water samples so that we can determine the fluctuations in water quality. So at each location, we're sending a meter through the water column, and at every meter depth, we're collecting pH, dissolved oxygen, temperature, chlorophyll, etc. At the surface and at the bottom of the water column, we're collecting nutrient samples. At the surface, we're also collecting toxin samples, and we're doing a qualitative analysis for different cyanobacteria species. On 2 dates, we are also collecting sediment samples along that transect, and the goal of that is to characterize legacy phosphorus concentrations. We're looking at both the sediment and the pore water. So this project is funded by state HAB funds, City of Stockton, Port of Stockton. In addition to those folks, Central Valley Water Board has been leading the sampling effort and boat time for this project. Next slide. Okay, so now I'm going to move into what are some spatial findings from each of those studies. Okay, so this first slide is the shellfish study, and we're looking at the microcystin concentration in water. This is over the entire sampling period, so from 2020 to 2022. On the x-axis, you see the 10 sampling locations, and then on the y-axis is the microcystin concentrations in microgram per liter. And the big takeaway here is that in these kind of more open areas of the delta, we're seeing relatively low microcystin concentrations. Below 5 micrograms per liter, this falls under that warning threshold of 6 micrograms per liter that the state uses. The other finding here is that we're seeing little to no microcystin at the Cache Slough sites and Sacramento River sites. Excellent. Okay, this is the same setup, but this time we're looking at the microcystin accumulation in Asian clams. So we have our x-axis the stations, and then the y-axis is the microcystin in nanograms per gram. That red line is EPA chronic exposure criteria. What that means is that the high-end consumers, the people that are eating the most shellfish, may have a greater risk if they're eating shellfish that are collected from sites that fall above that red line. So as you can see on this figure, the San Joaquin River at Turner Cut has by far the highest microcystin concentrations. And once again, at Cache Slough and most of those Sacramento River sites, we're seeing really a low concentration of microcystin in the shellfish. Moving on to the sediment study. So this is the microcystis seed stock in November. This is November 2020. We added an 8th station, so this is just 7 stations on the x-axis here in November 2020. And then on the y-axis is the total microcystis copies that were measured by qPCR. There were 5 samples taken at each location, and so they're very heterogeneous. We're seeing a lot of variation depending on where you grab that sediment sample. But what we do see is that Discovery Bay, Mildred Island, Stockton Waterfront, and Windmill Cove have the highest seed stock going into November. Stockton Waterfront was a little lower than we expected, and we think that this is an artifact of our sampling location. We were collecting right by the Morelli boat ramp, so there's a lot of boat traffic in and out that could be stirring up the sediment. So we switched our sampling for November 2021 and actually extended into the waterfront further and saw much higher concentrations of these resting cells in November 2021. Okay, next slide, please. This is moving on to the Stockton study now. So this is a single date. This is an isopleth from June 13th. On the x-axis is the transect that we're measuring. So up near Buckley Cove, Calaveras River, down to the public dock. On the y-axis, you see the depth of the water column. So the surface is at the top of the figure. And so moving across the transect, you can see that chlorophyll increases dramatically when you get on the other side of I-5 to that shallow portion of the Stockton waterfront. And this was the day this summer that we have measured the highest chlorophyll concentrations, and we measured up to about 22 micrograms per liter. I thought it was interesting that that chlorophyll, those high readings were throughout the entire water column. So not just the surface or the first meter or two, but you're seeing those high concentrations of chlorophyll down 4 meters depth. Very similar findings from each sampling event, this gradient of chlorophyll really increasing when you get to the shallower portion. There was no microcystin on this date. We didn't see that till later in the summer, but microcystin ends up being present at all of these sites, but increases along that same gradient as the chlorophyll. Now I'm going to do the same thing and move into more of the seasonal or temporal findings from each of these studies. Okay, so this is the shellfish study we're going to start with again. This time I've combined all of the stations and we're looking at the stations by month. Once again, that red line is that chronic exposure criteria. So what's safe to eat or not eat for these high-end consumers. And what you see is that the microcystin in the shellfish is most dominant in the July through November period. And in the winter months, we're not really seeing the microcystin in the shellfish, suggesting that it's leaving the shellfish. It's not accumulating and staying for a period of time. They're dissipating that toxin. Here's the sediment study. So on the left of November 2020, this matches up with the box plot that I shared with you earlier. But what I wanted you to see here is how the seed stock decreases quite a bit from November to April. So the high flows really flush out a lot of those resting cells, and you don't have as much seed stock when you move into April. However, Discovery Bay and Windmill Cove, both of these backwater areas, continue to have a relatively high seed stock to inoculate the spring or summer bloom. And I will say that April 2022, we also had high concentrations in the Stockton waterfront as well, probably based on, changing our sampling methods a bit. Okay, so this is another isopleth. We're back in Stockton now. This time, instead of spatially moving across a transect, we're now going, it's across time. So we're beginning in May, which was our first sampling date, and moving to August 24th, which is the last date that I've entered data for. So that peak chlorophyll, you'll remember that June 13th isopleth that I shared with you. You can see that here again on the graph. And the chlorophyll begins to drop after late June into early July. It still remains high, about 15 micrograms per liter. So interestingly, our first microcystin detection at this location was not until July 7th. So when we were out there, the bloom didn't look nearly as severe. The water looked a lot better, but that's when we were starting to get the toxin detection. By July 21st, we had our greatest microcystin concentration that we've measured to date, which is 59.1 micrograms per liter. So that exceeds the state danger threshold of 20 micrograms per liter. Microcystin is still present, but beginning to decrease by early August, even though the chlorophyll seems to kind of steady out from July into August. And I'll back up and saying, why are we seeing, high chlorophyll and not high toxin? This might be getting back to Keith's point. It could be related perhaps to dissolved inorganic nitrogen, which we haven't had the chance to analyze yet, or it could be a shift in toxin, non-toxin to toxin producing strains. Okay, another isoplet, this time we're looking at temperature. Once again, we're at the public dock, and I wanted to see how temperature changed across time. And what you can see is that July 21st is when we had peak temperatures, so we're 28, 29 degrees Celsius, and that corresponds to when we saw the highest toxin concentration. So the densest bloom, I wanted to grab that last point, occurred when water temperatures were cooler, 25 degrees Celsius, which is still relatively warm, but at this location was one of the cooler periods. So now moving on to the 3 data gaps. So the first data gap or knowledge gap that was identified to the Delta Nutrient Research Plan, which I thought our studies addressed, is where, when, and under what conditions do cyanobacteria blooms occur in the Delta. So location and timing play a key role. We're seeing the greatest densities of microcystis cells in these backwater areas. Dense microcystis blooms can occur and there can be no toxins present. We did do I didn't mention this, but we did a qualitative cell count and looked at the samples under the microscope, and that June 13th date when there was high chlorophyll, there was a lot of microcystis present. So microcystis was present then. In 2022, microcystin was greatest when we had the warmest temperatures, 27, 28 degrees Celsius. And microcystis seed banks are greatest in those backwater slough areas. The second research gap is what range and harmful algal toxins occur across different habitats. So although other toxins can be present in our studies, microcystin continued to be most prevalent. I should take in the delta off because Keith just explained some other toxins that he's found over the past year. But in Stockton, we were looking for cylindrospermopsin, anatoxin, and nodularin on all of these dates, and we've only detected microcystin. Outside of the dead-end sloughs and low velocity areas, microcystin concentrations appear to remain pretty low. They're remaining 5 micrograms per liter at the peak. And microcystins tend to be highest when temperatures are greater than 27, 28 degrees Celsius. And then the last knowledge gap is what is the status and trends for harmful algal toxins in fish tissue and bivalves? So we've identified microcystin concentrations in shellfish in areas of the delta where sturgeon may feed. We do know that sturgeon can feed on Asian clams. It's not the preferred food source, but there are fairly high concentrations. So this is an area that could be investigated a little bit more to see if this is indeed a stressor to sturgeon. We identified temporal pattern in microcystin accumulation in Asian clams. So we are seeing in the delta, the highest toxin concentrations in the shellfish seem to correspond with the bloom season. And then we're seeing spatial patterns in the microcystin accumulation. I didn't get into the crayfish data, but we're seeing our highest accumulation in both Asian clams and crayfish at the San Joaquin River at Turner Cup. Okay, and this is how can future research fill those same data gaps that I just went over. So where, when, and under what conditions do cyanobacteria blooms occur? Well, what do we know? We know that certain locations in the delta experience more severe blooms. We know these backwater habitats have the densest biomass, and they also seem to produce the highest toxin concentrations. We know that hotter and drier conditions typically result in the most severe blooms. So what do we need? We need to identify trends over time and space, which the previous speakers have also addressed. Further characterize conditions, including nutrients and habitats where cyanobacteria blooms are most prevalent. So we are doing that comprehensive nutrient portion of our study in Stockton, but I wasn't ready to present the data to you, but soon I will be. We need to prioritize and then characterize the areas that should receive mitigation. We can't treat the whole delta, but we could start focusing on some select locations. And we need to identify short and long-term control options feasible for application in the delta. A lot of mitigation out there for cyanobacteria is focused on lakes and enclosed water bodies. And so we need to figure out how do we translate some of those methods or come up with new methods so that we can address mitigation here in the delta. Filling these gaps will take both consistent monitoring, and then we also need to continue these special studies. Next slide. What range and harmful algal toxins occur across different delta habitats? What do we know? Well, in my studies that I presented today, microcystin was the most commonly detected cyanotoxin, but we have evidence that that's not entirely true. Microcystin can be present at low levels in a lot of delta locations. We have the highest microcystin concentrations in these dead-end sloughs. In June through November is when we're most likely to see microcystin present in multiple matrices. So what do we need? Continue to assess the different factors and drivers that are influencing toxin concentrations. How is climate change impacting these? Monitoring that over time will help inform that question. We need to analyze toxin in multiple matrices. The spat samples showed something different than the whole water samples. Similarly, the shellfish samples showed something different than the whole water samples. And then we need to prioritize areas to monitor for toxins. We can't monitor everywhere. So just some thoughts, we could focus on areas of greatest recreational exposure or areas of greatest exposure to sensitive species where managed species feeding throughout the delta, or we could go to areas where the toxins are measured as the highest. And then the last slide here. So what's the status and trends for harmful algal toxins in fish tissue? What do we know? We know microcystins are entering the food web. We've shown that filter feeders such as Asian clams and crayfish can accumulate these toxins. To date, cyanotoxins in the delta have not reached the concentration that have caused acute toxicity. What do we need? We need to prioritize the food web questions and knowledge gaps. There's a lot out there, so I think it would be helpful to figure out what we think are most important in the short term. Some ideas may be to determine potential impacts to the food web from chronic exposure. So what happens to a species that's, feeding on high toxin concentrations over a long period of time, and then examine organisms that feed in the food web for subchronic effects. All of this work could not have been done without the multiple partners that I've collaborated with on these projects. So I want to thank everyone on this slide. And then my questions.
Do Michael first.
Sort of a couple of related questions.
Yeah.
You mentioned, I think it was with the Stockton study, that you had legacy phosphorus versus, so my question is, what's the difference between legacy and non-legacy phosphorus? How does one distinguish between those? And you didn't talk about it, I know you didn't have very much time. But what is the rationale behind incorporating that measurement into that?
So for the purposes of that study, I'm thinking of legacy phosphorus as the phosphorus within the sediment that's bound to the metals that are in the sediment. So it's been entering the water column. That is kind of a measure of how much is in the area. The purpose is to start, before we do mitigation at any of these sites, we have to characterize them. So we can't just say, let's do this treatment without understanding how all the processes work at the site. So this is kind of like a first step. That's how I see it is. Let's do a really deep characterization of the site so we can start thinking about some potential mitigation options to help deal with the problem there.
I see it. I'll let someone speak.
Do you have questions on
I
that?
don't see any hands.
Okay. Anyone else in the room? Okay, Brian, and then.
No, I've already had my shot. Go ahead.
All right. I go up a lot of dead end sloughs. Not all dead end sloughs are the same. I mean, some of them are reliably, maybe this is faster than some of them you just don't see. And I'm just wondering if you have any ideas about why that would be, because we know that the organism is ubiquitous, and probably the spores are as well. But for all the things that we measure, there are no measurable differences. There are similar temperatures, similar nutrient concentrations, typically low nutrients.
Could be both activity possibly, maybe moving some of those seed stocks. I don't know, I'm just kind of throwing some ideas out. I don't know if we looked through the water column at how, things change. I think, are you mostly looking at the surface or are you looking at?
About 1 meter depth.
Okay, so perhaps there's changes, the chemistry between the sediments in the overlaying water column that's somehow affecting nutrient release into the water column. I don't know exactly what sloughs you're like thinking of that you don't see, because you're out there much more. Mine are in more locations than I've been. I've been more focused in Discovery Bay, Stockton, Windmill Cove, where we know we have problems. So what's like an example of a place where you
Like the stair step area of Liberty Island is a place where I'm not sure we ever see it. Up there, but we see all the other characteristics of a place we would expect to see it.
and the temperatures are warm,
The temperatures are very warm, the residence times are long. I guess I put that out there as an idea, and these sloughs are markedly different in their ability to produce.
like penetrate things to more turbid in some of them?
No, I'm just wondering if we have experiments out there that are natural experiments that we can explore. Yeah.
Yeah. Do you measure turbidity and
Oh,
at
yeah.
all those and they're all similar?
I just don't remember all my data off the top of my
Okay,
head.
yeah, because it seems like that could be a
That
factor.
could be a factor. Yeah.
All right, we're gonna wrap up because I want to let people get to lunch. Lunch is out. So for those of you participating virtually, we are gonna reconvene at 1:20. Very sharply at 1:20 because we're gonna stay on schedule today. And it looks like Jennifer, and for those of you in the room, it looks like Jennifer and Melissa have set up our lunch. So we will break for lunch until about 1:20. Any to say?
Yeah, questions until 12:35 and
Oh,
all
I'm
the
sorry.
previous.
Yeah, I'm sorry. Thank you. Sorry, we do have time for more questions, till 12:35, and then we will. Go ahead. Hi, so
Yeah, I don't know.
anyone in this session, if you have questions for anybody in this session, I guess I'll say.
Okay,
all right,
so. Well, I Dave had his hand up right before Meredith froze.
Yeah.
And then,
Me first.
And then Debbie, and then we'll go to if anybody else is online, and then we'll get Tom. Okay.
I'm not exactly, I don't know, well, the question came up while I was watching the presentation, and I don't know if that's you or just everybody, but I was curious, the plants are such an important sink for phytoplankton production or biomass. And has anyone done any calculations or based on filtering or heating preferences, whether it actually is an important sink from like persistent from the water column? So for example, you could have highest production rates, lowish steady state concentrations in the water because it's getting filtered rapidly and accumulating. I don't know if that's something that is at all feasible or plausible, or if that uptake is just more incidental.
Yeah, I don't know the exact answer to that, but it gets me thinking that could be another reason why we see the toxin in the bloom in some blues and not others, the presence of those clams. Something we've thought about as well.
Sometimes the studies ask whether a clam can distinguish between its food sources and just eject the cyanobacteria if it doesn't want to eat it, driving up the concentrations in a local area. And I don't believe we have studies showing that happens with Corbicula yet. But it's one of those questions as to whether selective feeding might change the local composition.
If no one jumps in yet, I'll ask another quick question. But I've had ballast water discharges on the Ray for the last several weeks, and I'm curious just about Stockton Ship Channel. And one of the things I read a recent paper showing that, I think it was, I exaggerate, something like a 2 to 4-fold increase in ballast water discharges to San Francisco Bay, San Francisco estuary ports since between 2004 and 2018 or something like that. And I originally was thinking of it more from the Bay perspective, but also Stockton Ship Channel would say that, is there, the timing of the big increases in microcystis in the Delta was in the early 2000s and thereafter. And not to say that temperature and other issues aren't the causes, but I wonder if that's been something that's been looked into this year.
Dave, can you repeat the question? People online are having a hard time hearing you. If you want to stand up when you're asking a question.
Move this to the back of the room.
Probably
so
why
that
not
people
work.
can- Put it back here. You all are talking a lot more.
It's all right,
Oh,
just,
sorry.
I'll move it,
I'll
I'll
walk
move
it
it
up.
around like a microphone.
I
I'll
was
move it
curious
around.
whether anybody knew if there's been any work looking at ballast water discharges and potential releases of cyanoHABs in this region.
Thinking perhaps, like, that's also where they're doing the greatest amount of dredging is where they're releasing that ballast water. So to me, I would think they'd be clearing those cells out with the dredging.
Interesting.
Idea.
I know Peggy's brought that up before.
All right,
I don't really remember what I have right now.
Isn't there a regulation though that indicates that they have to exchange their ballast water just outside because it's
So many miles off the coast.
there's so many miles and then they exchange that ballast water because it's a San Francisco Bay supposed to be one of the most invaded. And so that's how they're trying to cut that back. That doesn't mean that's still not a
do it. It's a program enforced by
An
the Coast
opportunity.
Guard.
That's what I thought too, but there's a clearinghouse of ballast water discharge data that shows that I guess there's a bit of a grace period for everybody to come into compliance, is what I understood. And I don't know if it's a freshwater issue or mostly marine issue, or sorry, estuarine.
So my questions were really just about a little bit about the monitoring styles. So when you were doing the spat sampling, how long is that your spat out for? Have you found some, or is it just a troll, or how are you doing that?
Yeah, so it's the same 2 weeks during the summer period, then it's a month during the longer. Yeah. And then for the map, I didn't actually show these data so much, but for the mapping, we have it adapted for our flow through system. So we put a spat in a small cooler and then pump the water through the cooler. And we mark when we put, like with a lat long, where we put in the spat, where we took it out. So we can also generate maps of where we picked up the spat data. And that was something that Rafe Cudella had done in the Bay program with Jim Klern. So we kind of adapted that method for the flow through mapping as well. And so so those are deployment lines.
Okay. And then as Ellen was talking about the PCR method, what method do you use and how does cost compare? Sorry.
No, you're good.
So the qPCR is looking at toxin or the genes. So you're not actually measuring the toxin itself. You're looking at the potential for toxin production. And then she also showed some data like using genes that only occur once in the cell. So you can use it as like a proxy for like the number of cells. Most of our work is on the chemical analyses. So we're actually looking at the molecules themselves. So and in terms of cost, the qPCR is cheaper than like running a chemical analysis per sample. But you don't actually get the actual toxin value. So
Toxin
you can't,
potential.
and a lot of like the objectives and regulations or advisory thresholds are for the toxins themselves.
The toxin genes can be present, but then there'd be There's not necessarily water or toxin in the water.
Yeah, and then the qPCR, yeah, like SPAT, qPCR tends to be more sensitive than the chemical test. So you can get a non-detect from your chemical test, but get like qPCR result or SPAT result often.
But also to add to that, one of the questions that we're trying to ask in our this monitoring studies is what is the difference between data you get from using LCMS analysis versus ELISA? I think that's something that came out of discussions with, we came and asked the community, what should we use, ELISA or LCMS? And it's not clear, so we're doing both. But what we've decided is, since we're looking kind of for presence absence, we started with running everything for LCMS, and then we run a subset for ELISA. So those are two completely different analytical approaches to detecting cyanotoxins. And so one of the things, once we have the fuller data set together, we're going to try and see how well those two analyses compare. So every time we collect discrete samples, we fill 2 bottles and we deploy 2 spats, and then we send them to the labs to do those parallel analyses. Okay. And yeah, and the cost on that is like, I don't know, it's like $500 a sample. Right? That's no.
Yeah.
Well, so do you guys have a sense of, now that we're seeing more toxins, does the ELISA end up to be cheaper? I mean, if we now have to look at Splendispermopsin, nodularin, there's like it's more than just persistence. So from a cost perspective, does it even save us money if we're using ELISA, which, there's lots of reasons to use different methods. We've got, on the marine side, that's been the age-old question, is which method do you use? And the answer is, it really depends on what your monitoring or research question is. Your methods have to give you the information that you need to answer your question. So that's a really important component, but historically, we've always thought of ELISA as being this cheaper option. And I gotta say, looking at the numbers these days and how many toxins we would have, just based on what you presented, Keith, I feel like we need to look at a wider range of toxins than we have been. So is it actually cheaper at the end of the day?
Yeah,
Do we know?
Yeah,
Sorry, I'm
no,
putting you on
no,
the
well,
spot.
I think it ultimately is kind of depend on like the labs you use and the costs. Like we.
Cause you guys get different rates.
Yeah, we're like, so we're,
Yeah.
so actually through some of our contracting and because we were doing everything, they did wind up being basically around equivalent. So it's not cheaper for us to do ELISA, but that was kind of set up to do the multiple analyses. So yeah, you're right in the sense that if you start stacking up all sorts of ELISA plates, it winds up being about the same. And then, yeah, we're running like 3 different LCMS methods to do all those. So for every sample, they kind of run it and then they tweak the parameters on the LCMS machine and then do the next one. So we could save some money. So yeah, it's like the microcystins, the nodularins, and the annabetapeptins are one, and then the anatoxins, saxitoxins. Are the other.
I would say it's much easier to do
Yeah.
ELISA.
With the ELISA, yeah, sure.
And so like,
Yeah.
if you did it in-house, you could do it so
Yeah.
much cheaper.
Yeah.
And you don't need a lot of equipment, you don't need a lot of
Yeah.
expertise to do
Yeah.
it, whereas
Yeah.
with LCMS, you need,
Yeah.
it's
that's
a very
true.
expensive instrument, it's a lot to pay to upkeep it, and you need a person that's basically trained in it to run it full time.
Yeah, that's true.
Okay, can we get a
Sorry,
couple
can
more
I ask
questions?
one more
Sorry.
question of Keith?
Okay.
So just based on what you're seeing now, where you're seeing more toxins than what has been detected historically, I think mostly because we haven't looked for all the toxins. Are you all at USGS planning to incorporate that into your future monitoring on a regular basis?
Well, so we're funded, I forgot to mention, so we're funded by the Delta Science Program, so thank you, Dylan. To do this through, I think it's the spring of 2024. So Delta Regional Monitoring Program helped start that effort, and then as well as some USGS funding, and then the last call. So yeah, we've got a couple more years of this.
So you've got a couple more years of doing all of those toxins.
At those stations, the mapping, incorporating the mapping is not funded beyond next year. So we were able to incorporate the toxins and are those, all those discrete mapping.
Okay.
But that, right,
We have
we have no fun, yeah, no mapping, period.
No mapping.
No mapping, period. Yeah, for next year. Yeah, and then, but then, yeah, through this, so until, yeah, so unless we get additional, yeah, so we have a couple more years with that.
Thanks. All right, Janis. Was there any other-
Our recording is expiring in 1 minute.
Okay.
Oh, okay.
Was there any that we had to get through?
Okay.
I didn't see any.
Okay, so quick, Tom, and then,
Keep
and
us
then
on
if
time.
people stay in the room, you know, you can catch the speakers.
Well, I just wanted Ellen to expound on the examples of mitigation that you were mentioning that might be used. And then is there any study where the effectiveness of mitigation? And I assume it's not nutrient controls, it's something else. Anyway, so
Well, I know Janis has been involved in potentially even peroxide treatment because
Looking at short term and long term, nothing specific. I mean, yes, definitely put nutrient in the long term category.
yeah.
So
Yeah.
are you aware of the city of Salinas has a city park with a lake? And they've spent an enormous amount of money trying to cap the sediments.
There are many failure stories. And
It's that's harder in the Delta.
Yeah,
to do like
anyway,
that.
so,
That's off the table one.
thank you everyone for the last session.